Hello and welcome to The Sleepy Evolutionist, where deep time meets tranquil dreams and evolution unfolds like a gentle lullaby across the vast cosmos. Tonight, we're drifting into the most fundamental story ever told, how water, simple H2O, became the architect of all life on Earth. It's a serene journey through 3.8 billion years of liquid magic that connects every drop in your body to the very first spark of life in ancient seas. Before we begin this quiet expedition through deep time, I'd love to know where in the world you're listening from and what time it is for you right now. Perhaps you're by an ocean, hearing waves that have carried the same water molecules for billions of years. Or maybe you're far inland, surrounded by the descendants of those primordial seas. No matter where you are, you're connected to this story in the most intimate way possible, through the water flowing through your very being at this moment. If you find this peaceful journey through liquid history soothing, I'd be ever so grateful if you gave this video a like and maybe even subscribed. It really helps this quiet corner of deep time contemplation grow, one dreaming soul at a time. Now, I do hope this isn't so fascinating that it keeps you awake. But if it does, well, at least you'll have learned something profound about the liquid foundation of your existence along the way. After all, understanding your watery heritage might just help you flow more peacefully into sleep. This video is for educational and relaxation purposes only, and is not intended as medical advice. So settle in, let your breathing find its natural rhythm like gentle waves on an ancient shore, and allow yourself to float through the grand chronicle of how water wrote the story of life itself. Let's begin our drift across the vast ocean of time. Let's drift gently into the very beginning, where water itself was born in the heart of dying stars. Picture, if you will, the vast cosmic nursery where hydrogen and oxygen first met in their eternal dance. This is not just chemistry. This is the prelude to your existence, written in the language of stellar fire and cosmic wind. In the immense furnaces of ancient stars, billions of years before our sun even dreamed of existing, oxygen was being forged from lighter elements through nuclear fusion. Imagine these stellar giants as cosmic alchemists, patiently crafting the very atoms that would one day flow through your bloodstream. Each oxygen atom that exists today was birthed in temperatures of millions of degrees, in stellar cores so dense that a teaspoon would weigh as much as a mountain. These were not violent explosions, but rather the gentle, persistent work of nuclear fusion, like a cosmic potter's wheel, spinning elements into existence across deep time. When these massive stars reached the end of their lives, they didn't simply disappear. Instead, they became generous grandparents to the cosmos, dispersing their precious cargo of newly made oxygen into the void through stellar winds, and eventually supernova. Think of this as the universe's way of seeding space with the raw materials for life itself. Your ancestors, in the deepest possible sense, were these dying stars, lovingly crafting and sharing the very substance that would one day become tears of joy, the morning dew, and the life-giving rain. Hydrogen, the universe's firstborn element, was already abundant from the Big Bang itself. In the cold depths of space, these hydrogen atoms drifted like cosmic snow, waiting patiently for their oxygen partners. When they finally met in the frigid molecular clouds between stars, they formed the first water molecules, not liquid, but ice crystals floating in the cosmic dark. These ice grains became the seeds around which everything else would grow, like tiny frozen libraries containing the potential for every ocean, every river, every drop of water that would ever exist. As our solar system began to form from a collapsing cloud of gas and dust about 4.6 billion years ago, water was already there, woven into the very fabric of creation. The young Earth, still glowing red-hot from its violent birth, seemed like the last place water could survive. Yet there it was, locked away in the rocks and minerals, waiting patiently for the planet to cool enough to reveal itself. Think of early Earth as a cosmic pressure cooker, with water vapor trapped in its superheated atmosphere, creating a blanket of steam that would eventually condense into the first rains. Your connection to this cosmic beginning runs deeper than you might imagine. Every water molecule in your body right now carries within it the signature of these stellar nurseries. The oxygen you breathe was forged in the heart of a star that died before our solar system was even born. The hydrogen bonding with it has been recycling through the cosmos for nearly 14 billion years, making each sip of water you take a communion with deep time itself. The delivery of water to Earth reads like a gentle cosmic love story, played out over hundreds of millions of years. Comets, those magnificent dirty snowballs from the outer reaches of the solar system, became the universe's postal service, carrying precious water ice on billion-year journeys to our young planet. Picture each comet as a message in a bottle, thrown by the cosmos itself, containing not just water, but the very possibility of life. These icy wanderers weren't rushing to their destination. Instead, they followed elegant elliptical paths, taking their time like thoughtful messengers who understood the precious nature of their cargo. When they finally arrived at Earth, they didn't crash destructively, but rather kissed our planet's atmosphere, their ice vaporizing and adding to the growing reservoir of water vapor that was already accumulating in the early atmosphere. It was as if the universe was carefully filling Earth's bathtub, one cosmic droplet at a time. But comets weren't the only delivery system. Asteroids, those rocky remnants of planetary formation, carried water locked within their mineral structures, like geological time capsules preserving cosmic moisture. Think of them as the universe's care packages, filled with hydrated minerals that would release their water when they gently impacted our cooling planet. This wasn't violent bombardment, but rather a patient, methodical process of accumulation, like rain filling a vast basin over countless seasons. The scientific precision of this delivery is breathtaking in its elegant simplicity. The ratio of deuterium to regular hydrogen in Earth's oceans tells us a story of mixed origins. Some water came from asteroids, some from comets, creating a blend that was just right for what was to come. It's as if the cosmos was following a recipe, carefully measuring ingredients from different sources to create the perfect medium for life. As millions of years passed, Earth's surface temperature finally dropped below the critical point where water vapor could condense into liquid. Imagine that first rainfall, not just a weather event, but the universe's inaugural celebration of liquid water on a planetary surface. These weren't ordinary rainstorms, but rather the cosmos weeping tears of joy, each droplet carrying within it the memory of stellar birth and death, of billion-year journeys through the cold void. The water that fell during those first primordial rains is the same water flowing through your body right now. When you feel thirsty and reach for a glass of water, you're participating in the same process that began in stellar cores billions of years ago. Your cells are cosmic museums, each one containing water molecules that have witnessed the birth of solar systems, the formation of planets, and now, miraculously, the emergence of consciousness capable of contemplating its own origins. Every time you cry, your tears connect you directly to that first cosmic rain. Every time you swim, you're floating in the same substance that once drifted between stars. The water in your morning shower has traveled further than any human explorer, carrying within it the patient wisdom of deep time and the gentle persistence of cosmic love. As millions of years drifted by like cosmic dreams, our planet transformed into something almost unimaginable, a world ocean, a sphere of liquid water floating in the darkness of space. Picture Earth 3. 8 billion years ago, not as the familiar continents and seas of today, but as Oceanus, a single unbroken body of water wrapping around a hidden rocky core like a sapphire blanket of possibility. This wasn't merely a large ocean. This was the universe's first attempt at creating a planetary womb. The water wasn't the clear blue of modern seas, but rather a rich, complex broth tinged with dissolved minerals from the rocky depths below. Iron gave it hints of rust red, while sulfur compounds painted it with yellows and greens. It was like looking at the palette from which life itself would be painted, each color representing a different chemical possibility waiting to unfold. The atmosphere above this primordial ocean was equally alien to our modern sensibilities. There was virtually no oxygen. The very gas you're breathing right now simply didn't exist in significant quantities. Instead, the air was thick with water vapor, carbon dioxide, methane, and ammonia. Think of it as the universe's first chemistry lab, where the atmosphere and ocean worked together as partners in the grandest experiment ever conceived, the creation of life from non-living matter. But here's what makes this story so deeply personal to you. Every cell in your body still carries the signature of this ancient ocean. The salt concentration in your blood is remarkably similar to that primordial sea, as if your circulatory system remembers its oceanic origins. When you taste your own tears or blood, you're literally tasting the ancient sea that gave birth to your most distant ancestors. Your body hasn't forgotten where it came from. It's maintained that chemical memory across nearly 4 billion years of evolution. The temperature of this global ocean was warm, but not scalding, heated gently by the young sun and by the radioactive decay of elements within Earth's core. Imagine it like a planetary hot spring, with temperatures ranging from merely warm to deliciously hot, creating gradients that would become crucial for the chemistry of life. These temperature differences weren't harsh or violent, but rather created gentle currents, like a slow, planetary-scale circulation system that mixed and stirred the chemical ingredients with infinite patience. Volcanic vents dotted the ocean floor, but rather than being destructive forces, they served as generous benefactors, steadily releasing minerals and energy into the water. Picture these as underwater springs of possibility, each one a library of chemical elements waiting to be read by the first living systems. They provided not just warmth, but also the chemical diversity that would become essential for life's emergence. The pressure at the bottom of this global ocean was immense, yet it wasn't crushing, but rather gentle, like being wrapped in a liquid embrace that held everything in perfect suspension. This pressure helped stabilize complex molecules that might otherwise have fallen apart, creating conditions where chemistry could experiment with increasingly sophisticated arrangements of atoms and molecules. Within this vast oceanic cradle, time moved with a different rhythm than anything we experience today. Days were shorter, only about 18 hours long, because Earth spun faster in its youth, like an eager child full of energy. The moon, our celestial companion, hung much closer in the sky, creating massive tides that swept across the planetary ocean twice each day with the regularity of a cosmic heartbeat. These weren't destructive forces, but rather gentle, rhythmic stirrings that mixed the ocean's chemistry, like a patient chef stirring an impossibly large pot of primordial soup. The lighting of this ancient world would have seemed almost dreamlike to modern eyes. Without continents to reflect sunlight, the entire planet glowed with the deep blue-green of infinite water, punctuated only by the occasional volcanic island rising briefly above the surface before being worn away by endless waves. Lightning crackled across the water-heavy atmosphere, but rather than being fearsome, each bolt was an artist's brushstroke, providing the electrical energy needed to forge complex molecules from simpler ones. Your connection to this world runs deeper than mere chemistry. The pH balance of your body fluids, the delicate acid-alkaline equilibrium that keeps you alive, is still calibrated to conditions that first emerged in that ancient ocean. When your kidneys work to maintain the perfect chemical balance in your blood, they're essentially maintaining an internal ocean environment that your ancestors have carried within themselves for billions of years. You are, in the most literal sense, a walking piece of that primordial sea. The waves of that ancient ocean didn't crash against beaches, because there were no beaches. Only endless water meeting endless sky in a horizon that curved unbroken around the entire planet. Yet these waves carried something precious, the first organic molecules, simple carbon-based compounds that formed when lightning, volcanic heat, and ultraviolet radiation work together to combine atmospheric gases with dissolved minerals. Think of each wave as carrying a library of molecular possibilities, gently rocking these early life precursors in a cosmic nursery. Beneath the surface, In the gentle darkness of the deep ocean, pressure and temperature created perfect conditions for what scientists call prebiotic chemistry, the quiet assembly of life's building blocks. Without oxygen to interfere, complex organic molecules could form and persist, slowly growing more sophisticated through billions of patient interactions. It was like watching the universe learn to write, one molecular letter at a time, eventually crafting the alphabet that would spell out your existence. The silence of this world would have been profound. No bird songs, no rustling leaves, no animal sounds of any kind. Just the gentle lapping of waves, the distant rumble of underwater volcanoes, and the soft patter of rain falling on an unbroken ocean surface. Yet this wasn't empty silence, but rather the quiet concentration of the universe engaged in its greatest work, the patient assembly of complexity from simplicity, the methodical construction of the foundations upon which all future life would build. When you float on your back in any body of water today, you're recreating that original state, your body naturally returning to the neutral buoyancy that your ancestors knew for their first billion years of existence. The feeling of weightlessness in water isn't just pleasant. It's your body remembering its original home in that vast, primordial sea that once wrapped around the entire planet like a liquid cocoon of infinite possibility. Let's pause for a moment to contemplate the quiet miracle of water itself. Those three atoms, arranged in such elegant simplicity that they become the foundation for all complexity. H2O seems so ordinary, so familiar, that we rarely stop to marvel at how extraordinary this molecule truly is. Yet within its humble structure lie properties so perfectly suited for life that it's as if the universe designed water specifically with biology in mind. The shape of a water molecule is beautifully bent, not straight, creating what chemists call a polar molecule, one end slightly positive, the other slightly negative, like a tiny molecular magnet. This isn't accidental geometry, but rather the key to water's extraordinary social nature. These molecular magnets attract each other with just the right strength, not so weak that water would fly apart as gas at Earth's temperatures, not so strong that it would lock into permanent ice. Instead, they create hydrogen bonds, gentle molecular handshakes that form and break billions of times per second in liquid water. Picture each water molecule as a sociable dancer at a cosmic ball, constantly changing partners, but never letting go completely. This molecular choreography creates water's most life-enabling property. It remains liquid across a range of temperatures that perfectly brackets the conditions where biological chemistry can flourish. Your body temperature of 98.6 degrees Fahrenheit isn't arbitrary. It sits right in the sweet spot, where water molecules dance with just the right amount of energy to support the chemical reactions that keep you alive. Water's ability to dissolve so many different substances earned it the nickname, the universal solvent. But this title barely captures its true importance. Think of water molecules as tiny molecular librarians, each one capable of surrounding and organizing other molecules in perfectly ordered arrangements. When salt dissolves in water, it doesn't disappear. Instead, water molecules lovingly embrace each ion, keeping them separated and mobile. This is how your blood can carry nutrients, hormones, and waste products to exactly where they need to go throughout your body. The density of water holds another quiet miracle. Unlike virtually every other substance on Earth, water becomes less dense when it freezes, not more dense. Ice floats because water molecules lock into a crystal structure that takes up more space than liquid water. This seemingly simple property had profound implications for early Earth. Ice formed an insulating layer on top of bodies of water, protecting the liquid below from freezing solid. Without this unusual behavior, Earth's early oceans might have frozen completely, ending life's story before it could begin. Your ancestors, swimming in those primordial seas, benefited from water's exceptional ability to store and transfer heat. Water has what scientists call a high specific heat capacity. It takes a lot of energy to change its temperature. This means that large bodies of water act like thermal stabilizers, absorbing heat when the environment warms and releasing it when temperatures drop. Early Earth's vast ocean was essentially a planetary thermostat, keeping temperatures stable enough for delicate chemical processes to unfold without being disrupted by sudden temperature swings. Water surface tension creates another layer of life-enabling magic. The hydrogen bonds between water molecules create an invisible skin on water's surface, strong enough to support insects, but gentle enough to allow easy penetration. In early Earth's ocean, this surface tension helped concentrate organic molecules, creating thin films where chemistry could experiment with higher concentrations of life's building blocks than would be possible in the bulk solution below. Perhaps most remarkably, water possesses what we might call molecular memory, its ability to form fleeting networks of hydrogen bonds that create temporary structures lasting mere trillionths of a second before reforming in new arrangements. These ephemeral networks give water properties that change subtly with temperature and pressure, making it not one substance, but thousands of slightly different versions of itself, each perfectly tuned to different biological needs. Your body today relies on water's extraordinary capacity for what scientists call hydrophobic effects. Water molecules are so strongly attracted to each other that they actually push oily, non-polar molecules together. This isn't rejection, but rather water's way of maintaining its own molecular society. This gentle expulsion force is what allows cell membranes to form, those crucial barriers that separate the inside of cells from the outside world. Without water's social preferences, the very concept of a cell, and therefore life itself, would be impossible. Water also serves as the universe's most patient chemical messenger. In liquid water, protons, single hydrogen nuclei, can hop from one water molecule to another with extraordinary ease, creating chains of chemical communication that span vast molecular distances in microseconds. This proton mobility is what makes acids and bases work, and it's the foundation for many of the chemical reactions that keep you alive. When you digest food, when your muscles contract, when your neurons fire, all of these processes depend on water's ability to ferry protons where they need to go. The quantum mechanical properties of water reveal even deeper layers of life-enabling magic. The hydrogen bonds that hold water molecules together are quantum mechanical phenomena, existing in multiple states simultaneously until they collapse into specific configurations when measured or disturbed. This quantum uncertainty gives water a fluidity that's perfectly calibrated for biological systems, rigid enough to maintain structure, flexible enough to allow constant change and adaptation. Your connection to these properties is intimate and immediate. The water in your brain right now exists in a state that's different from ordinary liquid water, organized around proteins and other biological molecules into structures that scientists are only beginning to understand. This biological water doesn't just fill space between brain cells. It participates actively in neural processing, helping to create the very consciousness that's contemplating these words. You are, quite literally, thinking with water. The isotopic composition of water, the rare varieties containing heavy hydrogen or heavy oxygen, tells stories of cosmic origin that your body preserves like a molecular archive. Every sip of water you take contains trace amounts of these rare isotopes, and your body incorporates them into its tissues. Years from now, scientists could analyze your hair or fingernails and determine something about the geographical history of the water you've consumed, creating a liquid autobiography written in isotopic signatures. Water's ability to remain liquid under pressure, becoming denser rather than changing phase, enabled life to explore the deep ocean environments where some scientists believe life may have first emerged. In the crushing depths around hydrothermal vents, water doesn't boil despite temperatures that would vaporize it at the surface. This high-pressure liquid water became a unique chemical environment where unusual reactions could occur, possibly including the very first steps toward living systems. Even water's expansion, when it freezes, serves life in ways that are still being discovered. As ice crystals form, they exclude dissolved substances, concentrating them in remaining liquid pockets. This freeze concentration effect may have been crucial for early life, creating high-concentration environments where complex chemistry could proceed much faster than in dilute solutions. Your ancestors may owe their existence to water's unusual behavior at its freezing point, making ice not an enemy of life, but a generous concentrator of possibility. In the gentle depths of that primordial ocean, chemistry began its first tentative experiments with complexity. Picture these early reactions not as violent collisions, but as delicate dances. Water molecules serving as both stage and choreographer for the universe's initial attempts at creating the molecular foundations of life. Each chemical interaction was like a whispered conversation in the liquid medium. Atoms and molecules finding new ways to arrange themselves in increasingly sophisticated patterns. The most fundamental reactions were surprisingly simple, yet profound. Carbon dioxide from the atmosphere dissolved readily in the warm ocean water, creating carbonic acid, a weak acid that nonetheless began the slow process of making the ocean's chemistry more complex. This wasn't dramatic acidification, but rather a gentle shift that allowed other reactions to proceed. When you drink carbonated water today, you're experiencing a mild version of what that ancient ocean tasted like. Slightly fizzy, slightly acidic, full of chemical potential waiting to unfold. Ammonia, abundant in the early atmosphere, dissolved into the ocean and began forming amino compounds, the nitrogen-containing molecules that would eventually become the building blocks of proteins. Think of these early reactions as the universe learning to spell with a new alphabet, adding nitrogen to its vocabulary of carbon, hydrogen, and oxygen. Your muscles, your enzymes, your DNA all trace their lineage back to these first tentative experiments with nitrogen chemistry in that ancient aquatic laboratory. Lightning provided the spark, literally, for many of these early reactions. Each electrical discharge that penetrated the ocean's surface carried enormous energy that could force atoms together in combinations that wouldn't normally occur. But this wasn't random destruction. The water medium ensured that only certain types of reactions could proceed, filtering the lightning's energy into productive channels. The amino acids in your body today may have their ultimate origin in lightning strikes that illuminated that ancient ocean billions of years ago. Ultraviolet radiation from the young sun, unfiltered by any ozone layer, penetrated deep into the ocean water and provided another source of chemical energy. But water served as both enabler and protector. The UV radiation could drive reactions near the surface. But as its energy was absorbed by water molecules, deeper layers remained gentle enough for delicate chemical structures to persist. This created a depth gradient of chemical activity, with high-energy reactions near the surface and more stable, complex chemistry in the peaceful depths below. Mineral grains suspended in the ocean water became tiny chemical factories. Clay particles, in particular, had surfaces that could organize simple molecules into more complex arrangements. Picture each grain of clay as a microscopic librarian, helping molecules line up in orderly sequences that would never occur in random solution. These mineral catalysts didn't just speed up reactions, they guided them toward specific products, showing the universe which molecular arrangements were most stable and useful. The dissolved metals in the ocean, iron, magnesium, zinc, and dozens of others, began forming coordination complexes with organic molecules, creating hybrid structures that were part mineral, part organic. These weren't alive in any sense we would recognize, but they possessed some lifelike properties. They could catalyze reactions. They could replicate their structures under the right conditions, and they could even evolve toward more efficient forms through simple chemical selection. Your blood contains iron atoms that may have first learned their chemical trade in these ancient metal-organic partnerships. As hundreds of millions of years drifted by like slow tides, the ocean's chemistry grew increasingly sophisticated through a process that can only be described as molecular education. Simple reactions taught the universe how to make more complex ones, each success building a foundation for the next level of chemical organization. Water wasn't just a passive medium for these reactions. It was an active participant. Its hydrogen bonding network providing scaffolding that stabilized intermediate compounds long enough for them to find more permanent arrangements. The formation of peptide bonds, the chemical links that connect amino acids into protein chains, may have first occurred in the gentle embrace of water molecules around mineral surfaces. These weren't the efficient, enzyme-catalyzed reactions that create proteins in your cells today, but rather slow, patient processes that required thousands of years to produce even short chains of amino acids. Yet each successful peptide bond was a triumph, a molecular achievement that brought the universe one step closer to creating systems capable of reproducing themselves. Phosphorus entered the chemical conversation through the slow weathering of rocks on the few volcanic islands that occasionally rose above the ocean surface. Phosphate compounds dissolved into the water and immediately began forming new types of chemical bonds, higher energy connections that could store and release energy in controlled ways. These phosphate groups would eventually become essential components of ATP, the energy currency of all living cells. The phosphorus atoms in your DNA and in every energy-carrying molecule in your body can trace their ancestry to those first phosphate solutions in the ancient ocean. Temperature gradients created by underwater volcanic activity established the first chemical engines. Regions where hot and cold water mixed, creating convection currents that could drive reactions forward that would otherwise reach equilibrium and stop. Think of these thermal gradients as the universe's first perpetual motion machines, using heat differences to keep chemistry moving in productive directions. Your body today still relies on similar principles, using temperature differences and pH gradients to drive the chemical reactions that keep you alive. The carbon chemistry that was developing in this aquatic medium was learning to create increasingly complex ring structures and chain molecules. Organic acids formed through reactions between carbon dioxide and simple hydrocarbons, creating the first molecules capable of donating protons in controlled ways. These early organic acids were the ancestors of the citric acid cycle that powers your cells today, an elegant series of reactions that extracts energy from food and makes it available for cellular work. Water's role as a hydrogen donor and acceptor became crucial as chemistry began, experimenting with oxidation and reduction reactions, processes that move electrons from one molecule to another. In the absence of free oxygen, these early redox reactions were gentler affairs, but they established the fundamental principles that would later power photosynthesis, respiration, and virtually every energy-producing process in living systems. When you breathe, when you digest food, when your muscles contract, you're using refined versions of redox chemistry that first developed in that ancient ocean. The ocean's vast volume provided something that laboratory chemistry lacks: time and space for rare events to become inevitable. Reactions that might have a 1 in a billion chance of occurring in any particular location became certain to happen somewhere when you had billions of cubic kilometers of water and millions of years of time. This is how the seemingly impossible became not just possible but inevitable, the patient mathematics of deep time, working with the generous expanse of the global ocean to make molecular miracles routine. Salt concentration gradients, created by the evaporation of water from shallow areas and its replenishment by deeper currents, established the first systems capable of concentrating and purifying chemical products. These natural distillation systems could take dilute solutions of organic compounds and concentrate them into more reactive forms, essentially creating the ocean's first chemical refineries. Your kidneys today work on similar principles, using concentration gradients to purify your blood and maintain the precise chemical balance that keeps your cells functioning properly. In the quiet depths of deep time, water became the universe's first chemistry teacher, gently guiding the formation of organic molecules with the patience of eons. These weren't complex substances by today's standards, but rather humble molecules that would become the alphabet from which life's poetry would eventually be written. Picture the ancient ocean as a vast, three-dimensional library where carbon atoms were learning to write their first tentative sentences in the language of organic chemistry. The simplest organic molecules formed through what scientists now call Miller-Urey chemistry, named after the researchers who first recreated these processes in the laboratory decades later. But in the real ancient ocean, these reactions proceeded with a gentleness and thoroughness that no laboratory could match. Methane gas bubbling up from the ocean floor met water molecules, energized by lightning or ultraviolet radiation, slowly forming formaldehyde, a simple molecule containing just one carbon atom bonded to hydrogen and oxygen. This humble compound would become a building block for more complex sugars, including the ribose that forms the backbone of RNA in your cells today. Water's role in these early syntheses was both protective and enabling. It provided a stable medium where delicate organic molecules could persist long enough to encounter other reactive partners. Yet it was also reactive enough to participate directly in bond-forming reactions. When formaldehyde molecules met in the aqueous medium, water molecules helped guide their condensation into longer-chain sugars through a process chemists call the Formose reaction. Each step was slow and patient, but the ocean had billions of years to perfect this molecular craftsmanship. Amino acids, the building blocks of proteins, formed through even more elegant chemistry. Simple compounds like hydrogen cyanide, ammonia, and formaldehyde, all dissolved in the ocean water, could combine under the gentle influence of heat and pressure to create glycine, alanine, and other simple amino acids. These reactions required no sophisticated catalysts, just the patient mediation of water molecules and the occasional input of energy from lightning or volcanic heat. The amino acids that form your muscles, your enzymes, and your brain were first synthesized in this gentle aquatic classroom billions of years ago. The ocean's varying conditions created different neighborhoods for different types of chemistry. In the cooler, deeper waters, molecules could form and persist without being broken apart by too much energy. In the warmer, shallower regions, higher energy levels drove more complex reactions, but also meant that only the most stable products survived. This created a natural selection process for molecular stability, a kind of chemical evolution that favored compounds capable of withstanding the ocean's various moods. Fatty acids began forming through the slow oxidation of simple hydrocarbons in the water. These long-chain molecules had an unusual property. One end loved water while the other end avoided it. This amphiphilic nature, loving and avoiding water simultaneously, would prove crucial for life's eventual development, as these molecules could spontaneously organize themselves into barrier structures that would become the first cell membranes. Your cell membranes today still rely on this ancient chemical property, using fatty acids to create boundaries between the inside and outside of every cell in your body. Nucleotide bases, the letters of the genetic code, also began their first tentative assembly in the aqueous medium. Purines like adenine could form from hydrogen cyanide in water, while pyrimidines like cytosine required slightly more complex reactions involving urea and other simple organic compounds. These bases had a natural affinity for each other that would later become the foundation of DNA as double helix structure. But even in these early stages, they showed preferential binding that hinted at the information storage systems that would eventually emerge. As these simple organic molecules accumulated in the ocean over millions of years, something remarkable began to happen. They started finding each other. Water's ability to organize molecules through hydrogen bonding created temporary clusters and associations, like molecular parties, where different compounds could meet and interact. These weren't permanent structures yet, but they were the first hints that chemistry was learning to create organized systems rather than just random collections of molecules. The concentration of organic molecules in the ancient ocean was probably quite low by modern standards. Imagine a weak tea rather than a rich broth. But in an ocean that covered the entire planet with no land to dilute it, even weak concentrations meant astronomical numbers of molecules in close proximity. Every cubic centimeter of that ancient water contained millions of organic compounds, each one a potential participant in the next stage of chemical evolution. Porphyrins, ring-shaped molecules that would eventually become the heart of chlorophyll and hemoglobin, began forming through the slow reaction of simple organic compounds with metal ions dissolved in the seawater. Iron atoms, abundant from underwater volcanic activity, found themselves surrounded by organic rings that could hold them in stable configurations. These early metalloporphyrins weren't alive, but they possessed some lifelike properties. They could catalyze chemical reactions. They could change their properties in response to their environment, and they could even participate in primitive electron transfer reactions. The iron in your blood today sits at the center of a porphyrin ring that's descended from these ancient molecules. Polymerization, the linking of small molecules into larger chains, began occurring spontaneously in certain regions of the ocean. Where minerals provided catalytic surfaces, amino acids could link into short protein-like chains, while nucleotides could connect into primitive RNA-like strands. These polymers were still far from being alive, but they represented chemistry's first experiments with information storage and retrieval. Some sequences were more stable than others. Some could catalyze their own formation, and a few could even make imperfect copies of themselves. The chirality puzzle, the fact that biological molecules come in specific left-handed or right-handed forms, may have been solved in this ancient aquatic environment. Water interacting with polarized light or with the magnetic fields generated by the Earth's rotation could have provided a subtle bias toward one molecular handedness over another. This seemingly minor preference would eventually become universal in living systems. Your amino acids are all left-handed. Your sugars are all right-handed. Patterns that may trace their origin to these earliest days of organic synthesis in the primordial ocean. Temperature cycling, caused by day-night variations and seasonal changes in the ancient Earth's climate, provided a natural mechanism for concentrating and purifying organic molecules. During warmer periods, water would evaporate from shallow areas, concentrating dissolved organic compounds. During cooler periods, fresh water would dilute these concentrations, but not before more complex reactions had time to occur in the concentrated solutions. This natural distillation process helped separate useful molecules from useless ones, creating a primitive form of chemical selection. The pH of different regions in the ancient ocean varied considerably, creating diverse chemical environments within the same planetary system. Acidic regions, created by dissolved carbon dioxide and organic acids, favored certain types of reactions, while more alkaline areas, influenced by dissolved minerals, promoted different chemistry. This pH diversity meant that the ocean functioned like a massive chemical laboratory with hundreds of different reaction chambers, each one exploring different pathways toward complexity. Hydrothermal circulation, the slow movement of water through heated rock formations, created another level of chemical sophistication. As ocean water percolated through hot volcanic rocks, it leached out dissolved minerals and underwent chemical transformations that were impossible at the surface. When this chemically altered water returned to the ocean, it brought with it new types of organic compounds and mineral-organic associations that added to the growing molecular diversity of the ancient seas. Your body's trace minerals, zinc, copper, molybdenum, and others may have first been incorporated into biological systems through these ancient hydrothermal processes. In the vast expanse of that ancient ocean, water was about to perform perhaps its greatest act of creativity, teaching molecules how to create boundaries, how to separate inside from outside, how to build the first walls that would define what it meant to be alive. This wasn't construction in any mechanical sense, but rather water's own social preferences, guiding molecules into arrangements that would change everything about how chemistry could work. The key players in this cosmic drama were amphiphilic molecules, compounds, with split personalities that reflected water's own complex nature. One end of these molecules was hydrophilic, loving water's polar embrace, while the other end was hydrophobic, preferring the company of oily, non-polar substances. Think of these molecules as diplomatic translators, able to communicate between water's hydrogen-bonded world and the realm of oily hydrocarbons that water couldn't directly touch. When these amphiphilic molecules, mainly fatty acids and similar compounds, synthesized through the ocean's patient chemistry, reached sufficient concentration, water's own behavior forced them into organized structures. Water molecules, so strongly attracted to each other through hydrogen bonding, essentially pushed the oily ends of these molecules together while embracing their water-loving ends. This wasn't rejection, but rather water's way of maintaining its own molecular society while accommodating these chemical immigrants. The result was spontaneous, by layers forming like molecular sandwiches, with water-loving heads facing outward toward the aqueous environment and oily tails hiding inside, protected from water's polar influence. These bilayers had an extraordinary property. They could close in on themselves, forming hollow spheres that created distinct inside and outside environments for the first time in Earth's history. Water had taught chemistry how to make rooms. Inside these first vesicles, these primitive cell-like structures, water behaved differently than it did in the open ocean. The confined space and the presence of the lipid membrane created a unique aqueous environment where molecules could accumulate to higher concentrations than would be possible in the infinite dilution of the ocean. Think of each vesicle as a tiny aquarium, maintaining its own special version of the ocean's chemistry. The permeability of these early membranes was beautifully calibrated by water's properties. Small molecules like water itself could pass through relatively easily, but larger organic molecules were retained inside the vesicles. This created the first example of selective permeability, the ability to control what enters and leaves a defined space. Your cell membranes today operate on exactly the same principles, using water's preferences to determine what can cross the boundary between your cell's interior and the outside world. Water's role in membrane formation was both architect and construction work. The hydrogen bonding network of water molecules provided the driving force that organized the amphiphilic molecules. But water also participated directly in the membrane structure. Water molecules became incorporated between the lipid molecules, creating a fluid, flexible boundary rather than a rigid wall. This water-mediated flexibility was crucial. It allowed the membranes to expand, contract, and even merge with other membranes without breaking apart. The size of these first vesicles was determined by the balance between water's surface tension and the membrane's mechanical properties. Too small, and surface tension would collapse them. Too large, and they would become mechanically unstable. The sweet spot was roughly the same size as modern bacterial cells, a few micrometers in diameter. Water had unconsciously designed the optimal size for the first living systems. These early vesicles created something unprecedented in the universe's chemical history, the first environments where evolution could occur in the modern sense. Inside each membrane-bound compartment, molecules could be concentrated, protected from dilution, and allowed to interact in ways that weren't possible in the vast ocean. Different vesicles could develop different internal chemistries, like separate evolutionary experiments running in parallel across the ancient seas. Water's behavior at the membrane interface created opportunities for energy storage and transfer that hadn't existed before. The slight electrical charge separation across the membrane, positive on one side, negative on the other, stored energy like a biological battery. Water molecules, with their own polar nature, could help transport charged particles across this barrier, creating the first primitive ion pumps and establishing concentration gradients that could be harnessed to do chemical work. The fusion and fission of these early vesicles, mediated entirely by water's properties, created the first mechanisms for sharing chemical information between separate compartments. When two vesicles approached each other, water molecules at the interface could reorganize the membrane lipids, allowing the compartments to merge temporarily and exchange contents before separating again. This was chemical sex before there was life, a way for different chemical systems to share their molecular innovations. Inside the vesicles, water assumed new roles it had never played in the open ocean. It became a structural component, helping to organize proteins and nucleic acids into functional shapes. It became a transport medium, carrying small molecules from one side of the compartment to the other. Most importantly, it became a reaction medium that was subtly different from bulk seawater, more organized, more concentrated, more capable of supporting the complex chemistry that life would require. The osmotic properties of water, its tendency to move across membranes to equalize concentrations, provided the first regulatory mechanism for these protocells. If the internal solution became too concentrated, water would flow in, causing the vesicle to swell. If it became too dilute, water would flow out, causing contraction. This automatic volume regulation, driven entirely by water's chemical properties, gave these early compartments a kind of primitive homeostasis, the ability to maintain stable internal conditions. Despite changes in the external environment, temperature variations in the ancient ocean created interesting effects on membrane behavior. Warmer water made membranes more fluid and permeable, allowing rapid exchange of molecules, but less stable compartmentalization. Cooler water made membranes more rigid and selective, creating more stable compartments, but slowing chemical exchange. This temperature sensitivity meant that different thermal zones in the ocean favored different types of vesicle behavior, creating diverse environments for chemical evolution. The pH buffering capacity of water within these confined spaces became crucial for maintaining stable internal conditions. Water molecules could absorb or release protons as needed, preventing the acidic or basic conditions that might destroy delicate organic molecules. This buffering ability, enhanced by the amphiphilic molecules of the membrane itself, created internal environments that were chemically more stable than the ocean outside. Your cell membranes today are direct descendants of these ancient water-organized structures. Every cell in your body maintains the same basic architecture, a lipid bilayer with water-loving heads facing the aqueous environments inside and outside the cell, and water-avoiding tails hidden in the membrane's interior. The water channels, ion pumps, and transport proteins embedded in your cell membranes all evolved from the basic principles that water established billions of years ago, when it first taught molecules how to create selective boundaries. When you think about cellular division, how one cell becomes two, you're witnessing the same membrane dynamics that first emerged in that ancient ocean. Water still provides the driving force, still organizes the lipid molecules, still determines the size and stability of the resulting compartments. The fundamental physics and chemistry haven't changed. They've just been refined and elaborated over billions of years of evolution. And so, in the gentle embrace of ancient water, chemistry finally learned its greatest lesson: how to become alive. This wasn't a sudden transformation, like lightning illuminating the darkness, but rather a gradual awakening, like the slow sunrise of consciousness itself. Water had spent hundreds of millions of years teaching molecules how to organize, how to cooperate, how to create boundaries. And now these lessons were about to culminate in something unprecedented: self-sustaining, self-reproducing chemical systems that we would recognize as the first life. Picture the moment when the first true cell drew its first breath, not literally breathing, of course, but performing that first act of metabolism that distinguished living chemistry from the complex but non-living chemical systems that had preceded it. This cell, perhaps no larger than a modern bacterium, floating in the warm waters of the ancient ocean, represented 4 billion years of patient chemical education finally paying off. Water had become midwife to life itself. These first organisms were prokaryotes, cells without nuclei, their genetic material floating freely in the aqueous interior like molecular libraries dissolved in liquid wisdom. Their DNA, that elegant double helix that carries the instructions for life, was stabilized and organized by water molecules that formed protective shells around each strand. The hydrogen bonds between water and the nucleotide bases helped maintain the precise base pairing that makes genetic replication possible A with T, G with C, held in place by water's molecular matchmaking. The cytoplasm of these first cells was essentially organized seawater, a concentrated aqueous solution containing proteins, nucleic acids, and small molecules, all suspended in water that had been specially modified by the cell's metabolic activities. This wasn't simply ocean water trapped inside a membrane, but rather water that had been transformed into a living medium, every molecule participating in the delicate chemical orchestration that sustained life. Protein synthesis, the process by which genetic instructions are translated into functional molecules, occurred entirely within this aqueous environment. Ribosomes, those molecular machines that read genetic code and assemble proteins, were themselves collections of proteins and RNA held together primarily by hydrogen bonds with water. The amino acids that formed proteins were delivered to the ribosomes by transfer RNA, molecules that navigated through the cell's watery interior like molecular postal workers, each one carrying its specific cargo to exactly the right destination. The first metabolic pathways were aqueous chemistry refined to extraordinary precision. These early cells could extract energy from simple chemical reactions, but unlike the random chemistry of the open ocean, their reactions were organized, controlled, and purposeful. Enzymes, proteins that catalyze specific reactions, provided the control, but water provided the medium in which these catalytic miracles could occur. Every enzyme active site was a precisely sculpted aqueous environment where water molecules participated actively in the catalytic process. Cell division, reproduction at its most fundamental level, was an aqueous ballet of extraordinary complexity. As the cell prepared to divide, its DNA was replicated by enzymes that moved along the double helix like molecular submarines, unwinding the strands and using water molecules to help attach new nucleotides in exactly the right sequence. The precision of this process, occurring entirely in an aqueous medium, ensured that genetic information could be passed from one generation to the next with remarkable fidelity. Your connection to these first cells is not just evolutionary, but literally chemical. The water in your cells today maintains the same basic chemistry as the water in those first prokaryotes. Your DNA is still stabilized by the same hydrogen bonding patterns with water molecules. Your proteins still fold into their functional shapes with the help of water's organizing influence. Your metabolic pathways still occur in aqueous solutions that are direct descendants of those first cellular interiors. These pioneering cells faced challenges that no living system had ever encountered before, and water proved to be not just their medium, but their most essential ally in solving each problem. Maintaining the right concentration of dissolved substances, what we now call osmotic balance, required these cells to develop sophisticated pump systems that could move water and dissolved ions across their membranes with exquisite precision. Your kidneys today perform similar functions, but using principles that were first established in these ancient single-celled pioneers. The genetic code itself, the universal language that translates DNA sequences into protein structures, was optimized for an aqueous environment. Each codon, each three-letter word in the genetic vocabulary, was designed to work in water-based systems. The redundancy built into the code, where multiple codons can specify the same amino acid, may have evolved as protection against the kinds of chemical errors that could occur in aqueous solutions. Your genes today still speak this ancient water-optimized language. Energy storage in these first cells relied heavily on phosphate compounds dissolved in the cellular water. ATP, adenosine triphosphate, became the universal energy currency because its high energy phosphate bonds were perfectly suited to the aqueous cellular environment. When ATP releases energy, it does so through hydrolysis, literally breaking bonds by adding water molecules. This fundamental energy process, powering every cell in your body right now, was established by those first aqueous life forms billions of years ago. The development of cell walls and other structural components occurred through processes that were intimately dependent on water's properties. Peptidoglycan, the mesh-like material that gives bacterial cell walls. Their strength is synthesized through reactions that occur in aqueous solution and require water molecules as both reactants and structural components. The flexibility and strength of these cell walls depend on their water content, creating structures that are rigid enough to maintain cell shape, but flexible enough to allow growth and division. Chemical communication between cells began almost immediately after the first organisms appeared, and water served as the medium for these earliest molecular conversations. Simple signaling molecules could diffuse through the aqueous environment, carrying information from one cell to another. This water-mediated communication allowed cells to coordinate their behavior, share resources, and even warn each other about environmental dangers. The neural signals in your brain today are descendants of these first aqueous chemical messages. The metabolism of these early cells was beautifully adapted to their aqueous environment. Glycolysis, the breakdown of glucose for energy, occurs entirely in aqueous solution and doesn't require oxygen. This ancient metabolic pathway, which your cells still use today as their primary energy extraction system, was perfected in those first organisms living in the oxygen-free waters of the early Earth. Every time you metabolize food, you're using biochemical machinery that was designed for an aqueous anaerobic world. The first mechanisms for DNA repair evolved in response to damage that could occur in aqueous environments, mainly the spontaneous chemical changes that water can cause in nucleotide bases over time. These repair systems had to work in aqueous solutions, using water-soluble enzymes to recognize damage, remove incorrect bases, and replace them with correct ones. Your cells today still use many of these same repair mechanisms, continuously fixing water-induced damage to maintain the integrity of your genetic information. Cell death, apoptosis, also had its origins in these early aquatic organisms. When cells became damaged beyond repair, they developed mechanisms to dismantle themselves in an orderly, controlled way that wouldn't harm neighboring cells. This cellular suicide process occurs through carefully regulated changes in the cell's water balance and the activation of water-soluble enzymes that break down cellular components. Even death, in these first living systems, was an aqueous process designed to protect the integrity of the cellular community. The emergence of sexual reproduction, the exchange of genetic material between different organisms, began with simple mechanisms that allowed cells to share DNA through aqueous transfer. Conjugation, transformation, and transduction, the three main ways that bacteria exchange genetic material, all occur through processes that depend entirely on the aqueous cellular environment. Your own sexual reproduction, despite its complexity, is based on these same fundamental principles of aqueous genetic exchange that were established in the very first living cells. In the vast aqueous theater of the ancient ocean, life began its greatest performance, learning how to change, how to improve, how to become something more than what it was before. Water served as both stage and script for this evolutionary drama, providing the medium in which genetic information could be copied, modified, and passed on to new generations. Every act of reproduction, every mutation, every evolutionary innovation that would follow was choreographed in the liquid embrace of aqueous chemistry. The copying of DNA during cell division required water's participation at every step. DNA polymerase enzymes, those molecular proofreaders responsible for replicating genetic information, worked by incorporating water molecules into their catalytic cycles. Each new nucleotide added to a growing DNA strand was attached through a reaction that released a water molecule, a process called condensation that created the phosphate sugar backbone of the double helix. The precision of this water-mediated copying process was extraordinary, with error rates so low that genetic information could be transmitted faithfully across millions of generations. But water also enabled the mistakes that would prove essential for evolution. Spontaneous hydrolysis, the random addition of water molecules to DNA bases, occasionally caused chemical changes that altered the genetic code. These weren't destructive errors, but rather opportunities for innovation, chances for cells to experiment with new genetic variations. The rate of these water-induced mutations was perfectly calibrated, frequent enough to drive evolutionary change, but rare enough to maintain genetic stability. Sexual reproduction, when it eventually evolved, transformed the aqueous cellular environment into a laboratory for genetic experimentation. The fusion of gametes occurred through the merger of their aqueous interiors. Combining genetic material in new ways, they created offspring with novel combinations of traits. Water served as the medium for this genetic mixing, allowing chromosomes to pair up, exchange segments, and separate into new combinations. Your own genetic uniqueness is the result of countless such aqueous genetic shuffles performed by your ancestors over billions of years. The development of more sophisticated reproduction systems required water's unique ability to organize complex molecular machines. The ribosome, that remarkable structure that translates genetic code into proteins, is essentially a water-organized ribonucleopride complex. Its active site, where amino acids are linked together to form proteins, is an aqueous pocket where water molecules participate directly in the peptide bond formation that creates new proteins. Without water's organizing influence, the ribosome couldn't function, and protein synthesis would be impossible. Horizontal gene transfer, the sharing of genetic material between different organisms, flourished in the aqueous environment of early Earth. Plasmids, small circular pieces of DNA, could move from one bacterial cell to another through aqueous channels, carrying with them genes for antibiotic resistance, metabolic capabilities, or other useful traits. This aqueous gene sharing accelerated evolution by allowing beneficial mutations to spread rapidly through bacterial populations, rather than being confined to single lineages. The evolution of more complex cellular structures occurred through water-mediated processes that gradually organized the cellular interior into specialized compartments. The development of organelles, those specialized structures within cells that perform specific functions, required the formation of internal membranes that created distinct aqueous environments within the same cell. Each organelle became a specialized aqueous laboratory where particular aspects of cellular chemistry could be optimized. Mutation repair systems also evolved in response to the kinds of damage that water could cause to genetic material. Cells developed sophisticated quality control mechanisms that could recognize water-damaged DNA, remove the damaged sections, and replace them with correct sequences. These repair systems had to work in the aqueous cellular environment, using water-soluble enzymes and cofactors to maintain genetic integrity. Your cells today still use many of these ancient repair mechanisms to protect your DNA from water-induced damage. The aqueous environment also became the testing ground for evolutionary innovations, with water itself serving as both judge and jury for which changes would prove beneficial. Proteins with new shapes and functions had to fold correctly in aqueous solution to be useful. Water's hydrogen bonding network provided the scaffolding that guided proper protein folding while rejecting configurations that wouldn't function properly. This water-mediated quality control assured that only genuinely useful evolutionary innovations would be preserved. Population genetics, the mathematics of how gene frequencies change over time, was fundamentally shaped by the properties of aqueous environments. The diffusion of molecules through water determined how quickly beneficial mutations could spread through populations, while the viscosity of water influenced how organisms could move and encounter potential mates. Even the concept of a generation was defined by how long it took for aqueous chemistry to complete a full cycle of DNA replication, cell growth, and division. The evolution of more complex reproduction strategies required increasingly sophisticated water management systems within cells. Meiosis, the special type of cell division that produces gametes, involves precise control of water movement across nuclear membranes and chromosome structures. The spindle fibers that move chromosomes during cell division are actually water-filled protein tubes that can extend and contract by regulating their water content. Your own reproductive cells still use these ancient water-based mechanisms to ensure proper genetic distribution. Chemical signaling systems evolved to coordinate reproduction across multiple organisms, and these communication networks relied entirely on water as their transmission medium. Pheromones, chemical signals that influence the behavior of other organisms, had to be soluble in the aqueous environments where early life existed. The sensitivity of these chemical communication systems was extraordinary. Cells could detect and respond to signaling. Molecules present in concentrations of just a few parts per billion in the surrounding water. The development of multicellularity, the evolution of organisms composed of many cooperating cells, required water-based coordination mechanisms that could synchronize the activities of thousands or millions of individual cells. Chemical gradients established in aqueous solutions provided positional information that told cells where they were in the developing organism and what type of tissue they should become. These morphogen gradients, diffusing through aqueous extracellular spaces, created the first developmental programs that could reliably build complex multicellular structures. Evolutionary arms races, the continuous competition between organisms with conflicting interests, played out in aqueous arenas where chemical warfare became increasingly sophisticated. Bacteria evolved the ability to produce antibiotics, water-soluble compounds that could kill competing organisms while simultaneously developing resistance mechanisms to protect themselves from the antibiotics produced by others. This aqueous chemical competition drove the evolution of increasingly complex biochemical pathways and sophisticated molecular recognition systems. The preservation of genetic diversity required water-based mechanisms for maintaining different versions of genes within populations. Balancing selection, frequency-dependent selection, and other evolutionary processes that maintain genetic variation all depend on the aqueous environments where organisms live and reproduce. The genetic diversity that exists in modern populations is the product of billions of years of water-mediated evolutionary processes. Your own reproductive system is a sophisticated collection of water-based mechanisms descended from these ancient aquatic innovations. Fertilization occurs when sperm cells swim through aqueous fluids to reach egg cells, using flagella that move by hydrolysis of ATP in aqueous solution. Embryonic development proceeds through carefully orchestrated changes in cell water content and the establishment of chemical gradients in aqueous environments. Even the production of reproductive hormones relies on enzyme systems that evolve to function in aqueous cellular environments. The genetic mechanisms that regulate reproduction, from the molecular switches that determine sex to the complex hormonal cascades that control reproductive timing, all evolved in aqueous systems and continue to function through water-based chemistry. When you think about having children, you're contemplating the activation of reproductive systems that have been refined over billions of years of aqueous evolution, systems that still depend fundamentally on the same water chemistry that enabled the first cells to divide and multiply in the ancient ocean. In the gentle depths of that ancient ocean, life was about to perform its most revolutionary act, learning how to eat sunlight. This wasn't merely a new way of obtaining energy. It was the invention of a process that would fundamentally transform both life and the planet itself. Water, having nurtured chemistry into biology, was now ready to become an active participant in the universe's most important energy conversion system, photosynthesis. Picture the first photosynthetic organisms, cyanobacteria floating in the warm, shallow seas of early Earth, as tiny solar collectors, each one a microscopic power plant capable of capturing light energy and converting it into chemical energy. But unlike modern solar panels, these biological systems required water not just as a coolant, but as an essential reactant, a source of both hydrogen atoms and the electrons needed to power the entire process. The chemistry of photosynthesis is fundamentally aqueous chemistry, occurring entirely within water-filled cellular compartments called thylakoids. Within these microscopic water-filled sacs, chlorophyll molecules, Those green pigments that capture light energy are suspended in carefully organized, aqueous solutions that optimize light absorption while preventing damaging photooxidation. The water surrounding each chlorophyll molecule isn't just a passive medium, but an active participant, helping to stabilize the pigment molecules and facilitating the rapid energy transfer that makes photosynthesis possible. The most revolutionary aspect of photosynthesis was its use of water as an electron donor, a source of the reducing power needed to convert carbon dioxide into organic compounds. Previous biological energy systems had used simpler molecules like hydrogen sulfide or organic acids as electron sources. But water was everywhere, unlimited, and constantly renewed by the planetary water cycle. This innovation freed life from dependence on scarce chemical resources and made the vast energy of sunlight accessible to living systems. The splitting of water molecules, photolysis, occurs at specialized protein complexes embedded in the thylakoid membranes. These molecular machines use light energy to break the strong covalent bonds holding water molecules together, releasing electrons, protons, and oxygen gas. This process, occurring billions of times per second in every photosynthetic cell, was biology's first venture into high-energy chemistry, requiring precise control to prevent the reactive products from destroying the cellular machinery. The oxygen gas released as a byproduct of water splitting was initially toxic to most living systems, including the cyanobacteria that produced it. These early photosynthetic organisms had to evolve sophisticated protective mechanisms to survive in the presence of their own waste product. They developed antioxidant systems, collections of enzymes and small molecules that could neutralize reactive oxygen species before they could damage cellular components. Your cells today still use many of these ancient antioxidant systems to protect against oxygen damage. The transformation of Earth's atmosphere from oxygen-free to oxygen-rich was gradual, occurring over hundreds of millions of years as photosynthetic organisms slowly accumulated in the ocean. Initially, the oxygen they produced was absorbed by dissolved iron and other reduced compounds in the seawater, creating the banded iron formations that we can still see in ancient rock strata today. These geological markers are literally the fossilized remains of ancient aquatic photosynthesis, evidence of the moment when life began changing the chemistry of the entire planet. Your connection to this ancient innovation runs deeper than you might imagine. Every breath you take contains oxygen molecules that were split from water by photosynthetic organisms. The hemoglobin in your blood, the protein that carries oxygen from your lungs to your tissues, evolved from much simpler molecules that originally helped early cells deal with the toxic effects of oxygen. Your entire aerobic metabolism is built on the foundation of aquatic photosynthesis that began billions of years ago in those ancient seas. The ecological impact of aquatic photosynthesis extended far beyond the organisms that performed it. As cyanobacteria multiplied in the ancient oceans, they created the first biological blooms, massive populations of photosynthetic organisms that could be seen from space, if there had been anyone to observe them. These blooms weren't chaotic explosions of growth, but rather organized communities where different species specialized in capturing different wavelengths of light, creating layered ecosystems that maximize the efficiency of solar energy capture. The carbon fixation process, the conversion of atmospheric carbon dioxide into organic compounds, occurred through a series of enzyme-catalyzed reactions that took place entirely in aqueous solution within the cells. The Calvin cycle, the biochemical pathway that accomplishes this transformation, requires a constant supply of water molecules, not just for the photolysis reaction, but also as participants in the various enzymatic reactions that build sugar molecules from carbon dioxide. Every sugar molecule in your body today was created through this ancient aqueous chemistry. Water's role in regulating photosynthetic efficiency was crucial for the success of early photosynthetic organisms. The fluidity of thylakoid membranes, controlled by their water content, determined how efficiently light energy could be captured and converted. too little water made the membranes too rigid for optimal function, while too much water diluted the pigment complexes and reduced efficiency. Evolution fine-tuned this water balance over millions of years, creating photosynthetic systems of extraordinary efficiency. The development of more sophisticated photosynthetic organisms led to the evolution of specialized aquatic ecosystems organized around light availability. Deep water species evolved pigments that could capture the blue-green light that penetrated furthest into the ocean, while surface species specialized in capturing the full spectrum of available sunlight. These depth-stratified communities created the first truly complex aquatic ecosystems, with energy flowing from photosynthetic primary producers to various levels of consumers. The seasonal cycles of photosynthetic activity, driven by changes in light availability and water temperature, established the first biological rhythms that would eventually become fundamental to all life on Earth. Circadian clocks, seasonal breeding cycles, and migration patterns all trace their origins to the rhythmic patterns of aquatic photosynthesis, established billions of years ago. Your own daily rhythms are descendants of these ancient photosynthetic cycles. The chemical byproducts of photosynthesis began to accumulate in the ocean water, creating new chemical environments that enabled the evolution of more complex metabolic pathways. Organic acids, sugars, and other photosynthetic products became food sources for non-photosynthetic organisms, establishing the first food webs based on primary productivity. The energy that powers your body today ultimately derives from photosynthetic processes that began in ancient aquatic environments. The evolution of sexual reproduction in photosynthetic organisms was facilitated by the aqueous medium in which they lived. Gametes could swim through the water to find mates, and genetic material could be exchanged through aqueous channels between cells. This sexual reproduction accelerated the evolution of photosynthetic efficiency by allowing beneficial mutations to be combined in new ways, leading to more sophisticated light harvesting systems and more efficient carbon fixation pathways. The symbiotic relationships that would eventually lead to the evolution of plant cells began in these ancient aquatic environments. Free-living cyanobacteria were engulfed by larger cells, but instead of being digested, they formed permanent partnerships with the cyanobacteria provided photosynthetic energy in exchange for protection and resources. These endosymbiotic relationships, mediated entirely by aqueous cellular environments, gave rise to the chloroplasts that power modern plant photosynthesis. Your dependence on photosynthesis extends beyond the oxygen you breathe to include virtually every molecule in your body. The carbon atoms in your proteins, nucleic acids, and lipids were all originally fixed from atmospheric carbon dioxide by aquatic photosynthetic organisms. Even the calcium in your bones and the iron in your blood were processed and concentrated by photosynthetic organisms before becoming part of the food webs that eventually led to your existence. You are, in a very real sense, made of concentrated sunlight and ancient seawater, assembled through billions of years of aquatic photosynthetic chemistry. As millions of years flowed by like gentle tides, the ancient ocean transformed from a simple chemical laboratory into a vibrant biological metropolis. Water, having taught chemistry how to become life, now served as the medium for life's first great creative explosion, a diversification of organisms, so remarkable that it would establish virtually all the fundamental body plans that still exist today. This wasn't chaos, but rather an elegant symphony of evolutionary experimentation, orchestrated entirely within the aqueous embrace of the sea. The Cambrian explosion, as modern scientists call this burst of biological creativity, occurred entirely in marine environments where water provided both the medium for life and the material for building new types of bodies. The first hard shells, the first compound eyes, the first jointed appendages, all of these innovations emerged from organisms, living and evolving in seawater that had been enriched by hundreds of millions of years of biological activity. Calcium carbonate shells, those protective armor systems that many marine organisms developed, formed through processes that were exquisitely dependent on water chemistry. Organisms learned to extract calcium and carbonate ions from seawater and organize them into crystalline structures using specialized proteins that could control crystal growth in aqueous solution. This biomineralization wasn't simply precipitation of minerals from solution, but rather a highly controlled biological process where water molecules participated actively in organizing and shaping the growing crystals. The evolution of complex sensory systems occurred in response to the unique properties of aquatic environments. Compound eyes, first developed by trilobites and other arthropods, were optimized for underwater vision, with specialized lenses that could focus light in the high refractive index medium of seawater. These eyes weren't just light detectors, but rather sophisticated optical instruments that could form clear images, detect motion, and even see polarized light patterns that were invisible to simpler visual systems. Filter feeding, the strategy of extracting food particles from water, became one of the dominant feeding strategies in the ancient ocean, leading to the evolution of increasingly sophisticated filtration systems. Sponges developed complex aquiferous systems that could pump thousands of times their own body volume of seawater through their tissues each day, extracting bacteria and organic particles with remarkable efficiency. The pumping mechanisms they used, based on the coordinated beating of flagellated cells, became the foundation for circulatory systems in more complex animals. Swimming, active movement through the aqueous medium, led to the evolution of diverse propulsion systems that took advantage of water's physical properties. Some organisms developed undulatory swimming, moving like aquatic snakes through the water. Others evolved jet propulsion, squirting water from their bodies to push themselves forward. Still others developed paddle-like appendages that could row through the water with remarkable efficiency. Each swimming strategy was perfectly adapted to the viscosity, density, and other physical properties of seawater. The development of predator-prey relationships in the ancient ocean drove an evolutionary arms race that produced increasingly sophisticated attack and defense mechanisms. Predators evolved sharp teeth, crushing claws, and venomous stingers, while their prey developed protective shells, camouflage, and early warning systems. All of these innovations had to work in aqueous environments, leading to solutions that were specifically adapted to underwater combat and defense. Your connection to this ancient explosion of creativity is written in your genetic code. The genes that control body plan development, the master control genes that determine where your head, limbs, and organs develop, are direct descendants of genes that first evolved in these ancient marine organisms. The basic toolkit for building complex multicellular bodies was assembled in the ocean, and your development still follows the same fundamental patterns established by your aquatic ancestors. Chemical communication systems became increasingly sophisticated as organisms learned to produce and detect signaling molecules in the aqueous medium, pheromones for attracting mates, alarm chemicals for warning of danger, and territorial markers for defending resources. All of these chemical messages had to be designed for aquatic transmission. The sensitivity of these communication systems was extraordinary. Some organisms could detect and respond to chemical signals present in concentrations of just a few molecules per billion in the surrounding seawater. The evolution of complex digestive systems allowed organisms to exploit new food sources and occupy new ecological niches. Digestive enzymes had to work in aqueous environments, often at different pH levels and salt concentrations than the surrounding seawater. Specialized digestive compartments evolved to create optimal aqueous conditions for breaking down different types of food, from plant material to animal proteins to mineral-rich shells and skeletons. Symbiotic relationships, partnerships between different species, flourished in the nutrient-rich waters of the ancient ocean. Cleaning stations where small organisms removed parasites from larger ones. Light-producing bacteria living inside translucent animals. Photosynthetic algae providing energy to non-photosynthetic hosts. These cooperative arrangements required sophisticated chemical communication and coordination all mediated by the aqueous environment they shared. The development of complex life cycles, including metamorphosis and alternation of generations, allowed organisms to exploit different aquatic environments during different stages of their lives. Larvae could disperse through the water column, while adults remained fixed to the sea floor. Reproductive stages could specialize in genetic exchange, while feeding stages focused on growth and energy storage. These complex life cycles required precise coordination of developmental programs with environmental conditions, all controlled by genetic and hormonal systems that operated in aqueous cellular environments. Biogeochemical cycles, the movement of chemical elements between living organisms and their environment, became established during this period of biological diversification. The carbon cycle, nitrogen cycle, and phosphorus cycle all developed as organisms learned to extract these essential elements from seawater, incorporate them into biological molecules, and return them to the environment when they died. These cycles created feedback loops between life and the planet's chemistry that continue to regulate Earth's environment today. The evolution of nervous systems allowed for increasingly complex behaviors and responses to environmental stimuli. Nerve cells had to transmit electrical signals through aqueous cellular environments, leading to the development of ion channels, neurotransmitters, and other components of neural signaling that still function in your nervous system today. The first brains were essentially concentrated collections of nerve cells optimized for processing sensory information in aquatic environments. Reproductive strategies became increasingly elaborate as organisms evolved ways to ensure successful mating and offspring survival in the vast ocean environment. Broadcast spawning, where organisms released eggs and sperm into the water for external fertilization, required precise timing and chemical coordination to ensure that gametes would find each other in the dilute aqueous medium. Internal fertilization evolved as an alternative strategy that provided more control over reproductive success, but required more complex anatomical and behavioral adaptations. The establishment of marine food webs created complex networks of energy and nutrient flow that connected organisms living at different depths and in different habitats throughout the ocean. Primary producers at the surface converted sunlight and dissolved nutrients into biological material, which then flowed through multiple trophic levels of consumers, decomposers, and recyclers. These food webs were held together by the aqueous medium that allowed dissolved nutrients to circulate throughout the system. Your body today reflects this ancient marine heritage in countless ways. Your blood plasma has a salt concentration similar to ancient seawater. Your neurons transmit signals using the same types of ion channels that evolved in marine organisms. Your digestive enzymes work optimally at pH levels that reflect their aquatic origins. Even your circadian rhythms are synchronized to tidal cycles that originated in the interaction between ocean waters and celestial mechanics. You are, quite literally, a terrestrial expression of ancient marine biology. In the flowing currents of ancient seas, water revealed another of its life-enabling talents, serving as the universe's most efficient delivery system, carrying nutrients, energy, and information to exactly where they were needed within living systems. This wasn't simple diffusion, but rather a sophisticated transportation network that would become the foundation for all the circulatory, digestive, and metabolic systems that sustain complex life today. Think of water as life's first postal service, with each molecule capable of carrying dissolved cargo across vast distances, from the depths of the ocean to the surface, from the outside of a cell to its interior, from one organ system to another. The efficiency of this aqueous transport system depended on water's remarkable ability to dissolve and carry an extraordinary variety of substances while maintaining their chemical integrity during transit. The movement of nutrients through water followed principles that would become fundamental to all biological transport systems. Concentration gradients, differences in the density of dissolved substances between different regions, created the driving force for movement, with nutrients naturally flowing from areas of high concentration to areas of low concentration. But living systems learned to harness and direct these flows, creating active transport systems that could move nutrients against concentration gradients when necessary. Glucose, the simple sugar that serves as life's most universal fuel, exemplifies how water-based transport systems enabled the evolution of complex metabolism. In single-celled organisms floating in the ancient ocean, glucose could be absorbed directly from the surrounding seawater through specialized transport proteins embedded in the cell membrane. These proteins worked like molecular turnstiles, allowing glucose molecules to pass through while excluding other substances. Your cells today still use essentially the same glucose transport mechanisms, refined over billions of years of evolution. The development of circulatory systems in more complex organisms was essentially an internal recreation of oceanic currents. Hearts evolved as biological pumps that could move aqueous solutions through networks of vessels, delivering nutrients and removing waste products with the same efficiency as ocean currents moving dissolved substances around the planet. Your cardiovascular system is a miniaturized version of the global ocean circulation that sustained the first living systems. Energy transport through aqueous systems became increasingly sophisticated as organisms evolved more complex metabolic pathways. ATP adenosine triphosphate emerged as the universal energy currency precisely because it was perfectly suited for aqueous transport. ATP molecules could dissolve in cellular water, travel to where energy was needed, release their stored energy through hydrolysis, reaction with water, and then be recycled back to high-energy form by metabolic processes that also occurred in aqueous solution. The buffering capacity of water became crucial for maintaining the chemical conditions necessary for efficient nutrient and energy transport. As organic acids and other metabolic byproducts accumulated in cellular water, buffering systems evolved to prevent harmful changes in pH that could disrupt transport processes. These buffering systems, combinations of weak acids and their salts dissolved in cellular water, maintained chemical stability while allowing the continuous flow of nutrients and energy through living systems. Enzyme systems that facilitate nutrient transport all evolved to function optimally in aqueous environments. These molecular machines had to recognize specific nutrients, bind them with just the right strength, and release them at exactly the right location, all while surrounded by water molecules that could interfere with these delicate interactions. The precision of these enzyme-mediated transport systems, still functioning in your body today, represents billions of years of evolutionary refinement in aqueous chemistry. The evolution of specialized transport organelles within cells created internal aqueous highways that could move specific types of cargo with remarkable efficiency. The endoplasmic reticulum became a network of water-filled channels that could transport proteins and lipids to their destinations within the cell. The Golgi apparatus evolved as a processing and shipping center, where proteins could be modified and packaged for transport in aqueous vesicles. These internal transport systems, all based on water-mediated movement, allowed cells to become increasingly complex while maintaining efficient nutrient and energy distribution. Osmotic regulation, the control of water movement across cell membranes, became essential for maintaining proper nutrient concentrations within cells. As nutrients were transported into cells, they increased the osmotic pressure of the cellular water, which could draw in excessive amounts of water and cause the cell to swell and burst. Organisms evolved sophisticated osmoregulatory mechanisms that could balance nutrient uptake with water management, maintaining optimal cellular chemistry while preventing osmotic catastrophe. The development of digestive systems created specialized, aqueous environments where complex nutrients could be broken down into simpler, transportable forms. Digestive enzymes worked in carefully controlled aqueous solutions where pH, salt concentration, and other chemical parameters were optimized for efficient nutrient processing. The stomach acid that helps you digest proteins today is a descendant of these ancient aqueous digestive systems, still using water-based chemistry to break down complex molecules into absorbable nutrients. Transport across biological membranes evolved, increasingly sophisticated mechanisms for moving nutrients and energy carriers between different aqueous compartments. Simple diffusion gave way to facilitated diffusion, where specific transport proteins could accelerate the movement of particular molecules. Active transport systems evolved that could use energy to move nutrients against concentration gradients, concentrating essential substances where they were needed most. Your kidney cells use these ancient active transport mechanisms to reclaim vital nutrients from your urine while eliminating waste products. The coordination of transport processes throughout complex organisms required the evolution of chemical signaling systems that could regulate nutrient flow in response to changing needs. Hormones, chemical messengers that could travel through aqueous body fluids to reach target tissues, evolved to coordinate metabolism, growth, and reproduction across multiple organ systems. Insulin, the hormone that regulates glucose transport into your cells, works through the same basic principles of aqueous chemical signaling that first evolved in ancient marine organisms. Waste removal became as important as nutrient delivery as organisms grew more complex and began producing toxic metabolic byproducts. The same aqueous transport systems that delivered nutrients could also carry away waste products, but this required mechanisms for distinguishing between useful and harmful substances. Filtration systems evolved that could selectively remove waste while retaining valuable nutrients, leading to the development of excretory organs that still function in your body today. The storage of nutrients and energy in aqueous systems led to the evolution of specialized tissues and organs that could serve as biological warehouses. Glycogen, the stored form of glucose in animal cells, accumulates in aqueous cellular environments where it can be quickly mobilized when energy is needed. Fat storage requires specialized cells that can manage the interface between aqueous cellular environments and hydrophobic lipid droplets. Your liver, muscles, and adipose tissue today all use storage mechanisms that evolved from these ancient aqueous transport and storage systems. Energy coupling, the ability to use energy released from one reaction to drive another reaction that requires energy input, became possible through aqueous intermediates that could carry chemical energy from where it was produced to where it was needed. The phosphorylation of ADP to form ATP, powered by energy from glucose metabolism, occurs entirely in aqueous solution and allows cells to store energy in a form that can be easily transported and used throughout the organism. Seasonal and circadian regulation of transport processes allowed organisms to optimize nutrient and energy management in response to changing environmental conditions. Transport rates could be increased when nutrients were abundant and decreased when they were scarce. Energy storage could be enhanced in preparation for periods of low food availability. These regulatory mechanisms, controlled by genetic and hormonal systems that operate in aqueous cellular environments, still govern the metabolism and energy management of your body today. After nearly 3 billion years of aquatic evolution, life faced its greatest challenge and opportunity, learning to survive outside the liquid embrace that had nurtured it since the beginning. This migration from water to land wasn't an abandonment of aquatic life, but rather an expansion of it, as organisms carried their essential water chemistry with them into new environments. Every step of this transition required innovative solutions for maintaining the aqueous internal environments that life had come to depend upon. Picture the first tentative explorers, perhaps algae in tidal pools or primitive fungi at the water's edge, discovering that the interface between aquatic and terrestrial environments offered unique advantages. These marginal habitats provided access to abundant sunlight without the filtering effects of deep water, while still maintaining enough moisture to support aqueous cellular chemistry. Success in these borderland environments required organisms to become living aquariums, maintaining their own internal oceans while venturing into the dry world beyond. The evolution of protective barriers against water loss became the first priority for terrestrial colonizers. Waxy cuticles, waterproof cell walls, and sealed body surfaces all served the same function, preventing the evaporation of precious internal water while still allowing necessary gas exchange with the atmosphere. These weren't just protective coatings, but rather sophisticated interfaces that could regulate water loss while maintaining the chemical exchanges necessary for life. Root systems and other water harvesting structures evolved as biological mining operations, designed to extract water from soil and concentrate it within living tissues. These underground networks weren't just passive absorbers, but active water pumps that could generate surprising amounts of pressure to move water upward against gravity. The tallest trees today still use refined versions of these ancient water transport systems, moving water from their roots to their highest branches through purely aqueous mechanisms. The development of internal circulatory systems became essential for distributing water and dissolved nutrients throughout increasingly large and complex terrestrial organisms. Hearts, blood vessels, and lymphatic systems all evolved as ways to maintain aqueous transport networks within bodies that were no longer immersed in water. Your cardiovascular system today is essentially a closed-loop ocean, circulating the same salty aqueous solution through your body that your ancestors once swam in. Respiratory systems evolved to extract oxygen from air while minimizing water loss through evaporation. Lungs, gills, and other gas exchange surfaces had to remain moist to function properly, but they also represented major sites of potential water loss. The evolution of efficient respiratory systems required sophisticated compromises between gas exchange efficiency and water conservation, leading to the complex breathing mechanisms that keep your cells supplied with oxygen while preserving your body's water content. The colonization of terrestrial environments created new opportunities for diversification as organisms adapted to different moisture conditions. From desert environments where water conservation was paramount to tropical rainforests where abundant moisture allowed for more extravagant water use. Each environment selected for different water management strategies, leading to the incredible diversity of terrestrial life forms we see today. Reproductive strategies had to be completely reimagined for terrestrial environments, as gametes could no longer rely on surrounding water for transport and protection. Internal fertilization evolved as a way to provide aqueous environments for sperm and egg cells even in dry conditions. Protective shells, amniotic fluid, and other innovations created portable aquatic environments where embryos could develop using the same water-based chemistry that had served their aquatic ancestors. The sensory systems that had evolved for aquatic environments required major modifications to function in air, a medium with very different physical properties than water. Vision had to adapt to the lower refractive index of air compared to water, leading to the evolution of new types of lenses and corneas. Hearing systems evolved to detect sound waves traveling through air rather than water, requiring new mechanisms for amplifying and processing acoustic signals. Chemical sensing smell and taste had to work with molecules dissolved in minimal amounts of water rather than in abundant aqueous solutions. Thermoregulation became a new challenge as organisms left the thermally stable environment of large water bodies for the more variable temperatures of terrestrial environments. Water's high heat capacity, which had provided thermal buffering in aquatic environments, now had to be managed within the confines of individual organisms. Sweating, panting, and other evaporative cooling mechanisms evolved to use water's latent heat of vaporization for temperature control, while behavioral adaptations helped organisms avoid excessive water loss during temperature regulation. The evolution of complex terrestrial ecosystems recreated many of the nutrient cycling patterns that had first developed in aquatic environments. Soil became an aqueous medium where dissolved minerals could be processed by plant roots, fungi, and bacteria. Rivers and streams served as terrestrial circulatory systems, carrying nutrients from highlands to lowlands and eventually back to the oceans. The global water cycle became the mechanism that connected terrestrial and aquatic ecosystems into a single planet-wide system. Symbiotic relationships between organisms became even more important in terrestrial environments, where the challenges of water and nutrient acquisition were greater than in aquatic systems. Mycorrhizal fungi formed partnerships with plant roots, extending their water and nutrient-gathering capabilities. Nitrogen-fixing bacteria in root nodules provided essential nutrients in exchange for sugars and other organic compounds. These partnerships were mediated entirely by aqueous chemical communication systems that had evolved in marine environments. The development of seeds and other reproductive structures allowed terrestrial organisms to survive periods of drought while maintaining the aqueous chemistry necessary for life. Seeds contained concentrated aqueous solutions that could remain viable for months or years, then rapidly rehydrate and resume metabolic activity when water became available. Spores, cysts, and other dormant stages all used similar strategies for preserving aqueous cellular chemistry during unfavorable conditions. Social behaviors in terrestrial animals often centered around water sources, with complex territorial and cooperative behaviors evolving around the need to secure and share access to this essential resource. Migration patterns, seasonal movements, and habitat selection all reflected the fundamental requirement for maintaining adequate water intake while avoiding environments where water loss would be excessive. Your own preference for living near water sources reflects this ancient terrestrial adaptation. The evolution of kidneys and other excretory organs became crucial for terrestrial life, as organisms could no longer simply release waste products into surrounding water. These organs had to concentrate waste products while conserving precious water, creating concentrated urine and other excretory products that minimized water loss while eliminating toxic substances. Your kidneys today can produce urine that's far more concentrated than your blood plasma, a remarkable feat of aqueous chemistry that allows you to survive in relatively dry environments. The development of internal skeletons and other structural support systems compensated for the loss of buoyancy that organisms had enjoyed in aquatic environments. Bones, cartilage, and other skeletal materials had to provide structural support while still allowing for the circulation of aqueous body fluids. Your skeleton today is not just a framework, but also a reservoir of minerals that can be mobilized when needed, and a structure that's constantly being remodeled by cells that operate in aqueous tissue environments. Wound healing and immune responses in terrestrial organisms had to work without the cleansing and diluting effects of surrounding water. Specialized aqueous solutions, blood, lymph, and tissue fluids evolved to serve these functions, creating internal aquatic environments where immune cells could operate and wounds could heal using the same basic mechanisms that had evolved in marine organisms. Your immune system today is essentially an internal ocean where specialized cells patrol for threats and coordinate defensive responses through aqueous chemical communication. In the flowing depths of evolutionary time, life embarked on one of its most ingenious innovations, creating internal rivers, aqueous highways that could transport the essence of life to every corner of increasingly complex organisms. The evolution of circulatory systems was essentially the invention of portable oceans, allowing organisms to carry the benefits of aquatic chemistry within their own bodies as they grew larger and more sophisticated. Think of the first primitive circulatory systems as life's attempt to solve a fundamental problem: how to maintain the intimate contact between cells and their aqueous environment that had been possible when organisms were simple, single-celled creatures floating in the ocean. As multicellular organisms grew larger, their interior cells found themselves increasingly isolated from the life-giving properties of surrounding water. The solution was revolutionary: bring the ocean inside. The earliest circulatory systems were beautifully simple: open cavities where aqueous body fluid could slosh around, bathing internal organs directly in a solution that was essentially modified seawater. These hemolymph systems, still used by many invertebrates today, created internal aquatic environments where dissolved nutrients, oxygen, and waste products could move by simple diffusion and gentle circulation. Your distant arthropod cousins, insects, crabs, and spiders still use refined versions of these ancient open circulatory systems. The evolution of closed circulatory systems represented an extraordinary advancement in aqueous engineering. The creation of sealed networks of vessels that could direct the flow of internal body fluids with unprecedented precision. Hearts evolved as biological pumps, remarkably similar in principle to the mechanical pumps that move water through modern plumbing systems, but crafted from living tissue and powered by the same ATP chemistry that energizes all cellular processes. The chemistry of blood, that remarkable aqueous solution that serves as your body's internal ocean, preserves the signature of ancient seawater while adding sophisticated modern capabilities. The salt concentration of your blood plasma is strikingly similar to the salinity of ancient seas, as if your circulatory system remembers its oceanic origins. But blood is far more than modified seawater. It's a complex aqueous solution containing hundreds of dissolved substances, each one precisely regulated to maintain optimal conditions for cellular life. Hemoglobin, the iron-containing protein that carries oxygen in your blood, represents one of evolution's most elegant solutions to the challenge of oxygen transport in aqueous systems. This remarkable molecule can bind oxygen in the high oxygen environment of your lungs and release it in the low oxygen environment of your tissues, all through subtle changes in its molecular shape that are mediated by the aqueous environment of your blood plasma. The iron atoms at the heart of each hemoglobin molecule are descendants of the metalloporphyrin complexes that first formed in the ancient ocean billions of years ago. The development of lymphatic systems created a secondary, aqueous circulation that could collect fluid that leaked from blood vessels and return it to the main circulatory system. This lymphatic circulation also became the highway for immune system cells, creating an internal aqueous environment where specialized immune cells could patrol for threats and coordinate defensive responses. Your immune system essentially operates as a mobile aqueous laboratory, with white blood cells carrying sophisticated chemical detection and response systems throughout your body's internal ocean. The evolution of specialized circulatory adaptations allowed different organisms to thrive in diverse environments while maintaining their essential aqueous internal chemistry. Arctic fish developed antifreeze proteins that prevent ice crystal formation in their blood, allowing them to remain active in water that would freeze the blood of most other animals. Desert mammals evolved highly efficient circulatory systems that minimize water loss, while maintaining adequate circulation to all body tissues. The regulation of blood pressure and flow became increasingly sophisticated as circulatory systems evolved to serve larger and more complex organisms. Smooth muscle cells and blood vessel walls learned to contract and relax in response to chemical signals dissolved in the blood, creating a dynamic system that could redirect blood flow to where it was needed most. During exercise, your muscles receive increased blood flow through the same basic mechanisms that first evolved in ancient marine organisms: chemical signaling through aqueous solutions. Capillary networks, those microscopic vessels that bring blood into intimate contact with individual cells, represent the circulatory system's attempt to recreate the direct cell-to-environment contact that existed when life was still single-celled and surrounded by ocean water. The walls of capillaries are so thin that dissolved substances can pass directly between blood and tissue fluid, essentially bathing every cell in your body in a carefully regulated aqueous environment that maintains optimal conditions for cellular metabolism. The evolution of portal circulation systems created specialized aqueous highways that could carry chemical signals directly between organs. The hypothalamic pituitary portal system, which connects your brain to your hormone-producing pituitary gland, allows for precise chemical communication through aqueous transport. Similarly, the hepatic portal system carries nutrients absorbed from your intestines directly to your liver for processing, using the same principles of aqueous transport that first evolved in marine organisms. Thermoregulation through circulatory systems became a crucial innovation that allowed organisms to maintain optimal body temperatures while preserving water balance. Vasodilation, the expansion of blood vessels near the body surface, increases heat loss by bringing warm blood closer to the environment, while vasoconstriction conserves heat by reducing blood flow to the skin. These responses are controlled by chemical signals dissolved in the blood and represent sophisticated uses of aqueous chemistry for temperature management. The development of contracurrent exchange systems and circulatory networks allowed for remarkable efficiency in heat and substance conservation. In the extremities of Arctic animals, arteries and veins run close together, allowing warm arterial blood to transfer heat to cool venous blood before it returns to the heart. Fish gills use similar contracurrent arrangements to maximize oxygen extraction from water. These systems represent sophisticated applications of aqueous chemistry and physics that evolved to optimize resource use in challenging environments. Blood clotting mechanisms evolved as emergency response systems that could quickly seal damaged blood vessels while maintaining circulation to the rest of the body. The cascade of chemical reactions that produces blood clots occurs entirely in aqueous solution and requires precise coordination of multiple proteins and cellular components. This system had to be carefully balanced, responsive enough to quickly stop bleeding, but controlled enough to prevent inappropriate clot formation that could block circulation. The evolution of high-performance circulatory systems enabled the development of warm-blooded metabolism, which requires extremely efficient oxygen and nutrient delivery to support the high metabolic rates needed for internal heat production. Birds and mammals developed four-chambered hearts that completely separate oxygenated and deoxygenated blood, creating highly efficient aqueous transport systems that can support the energy demands of flight, complex behaviors, and large brains. Specialized circulatory adaptations for extreme environments demonstrate the versatility of water-based transport systems. Deep-diving mammals evolved enlarged blood volumes and specialized oxygen storage systems that allow them to remain underwater for extended periods. High-altitude organisms developed increased numbers of red blood cells and enhanced oxygen-carrying capacity to function in low-oxygen environments. These adaptations all work through modifications of basic aqueous chemistry principles. Your cardiovascular health today depends on maintaining the same basic aqueous chemistry that evolved billions of years ago. In marine organisms, the balance of electrolytes in your blood, the pH buffering that prevents dangerous acidity or alkalinity, the osmotic regulation that maintains proper cell hydration. All of these life-sustaining processes use mechanisms that trace their origins to the ancient ocean. When you drink water, exercise to improve circulation, or take medications that affect blood chemistry, you're participating in the ongoing maintenance of an internal aquatic ecosystem that connects you directly to the primordial seas where life began. Across the vast sweep of evolutionary time, water orchestrated perhaps its most magnificent symphony, the development of organisms composed of trillions of cooperating cells, each one a specialized citizen in biological societies of breathtaking complexity. This transition from single cells to multicellular organisms wasn't simply a matter of cells sticking together, but rather the invention of entirely new forms of aqueous organization that could coordinate the activities of countless individual units into coherent, functioning wholes. Picture the first tentative experiments in multicellularity, perhaps colonies of cells that remained connected after division, sharing resources through aqueous channels that linked their cytoplasms. These early cellular cooperatives discovered that working together in aqueous environments offered advantages that no single cell could achieve alone. Larger size meant less vulnerability to predation. Specialized cell types could perform specific functions more efficiently, and cooperative behavior could exploit resources that were inaccessible to individual cells. The evolution of cell-to-cell communication systems required the development of sophisticated chemical signaling networks that could coordinate activities across multiple cells. These early signaling molecules had to be soluble in aqueous intercellular fluids and capable of carrying specific information from one cell to another. Gap junctions evolved as direct aqueous connections between adjacent cells, allowing the rapid exchange of small molecules and electrical signals that could synchronize cellular activities across entire tissue sheets. Differentiation, the process by which identical cells develop into specialized cell types, occurred through carefully orchestrated changes in gene expression that were controlled by chemical signals dissolved in the aqueous cellular environment. Master control genes could be activated or suppressed by signaling molecules that bound to specific DNA sequences, creating cellular decision points where individual cells committed to particular developmental pathways. Your body today contains hundreds of different cell types, each one specialized for specific functions, but all derived from the same original fertilized egg through these ancient aqueous differentiation processes. The development of extracellular matrix, the aqueous gel-like substance that surrounds and supports cells in multicellular organisms, created structured environments where cells could organize into tissues and organs. Collagen, the most abundant protein in your body, forms networks of fibers embedded in aqueous gels that provide mechanical support while allowing for the diffusion of nutrients and signaling molecules. This extracellular aqueous environment became a medium for cellular communication and coordination that was far more sophisticated than the simple seawater that surrounded single-celled organisms. Morphogenesis, the process by which developing organisms acquire their characteristic shapes, required precise control of cell division, cell movement, and cell death in aqueous environments. Chemical gradients established in tissue fluids provided positional information that told cells where they were in the developing organism and what structures they should form. These morphogen gradients, diffusing through aqueous extracellular spaces, created the first biological GPS systems that could guide cellular development with remarkable precision. The evolution of organ systems required the coordination of multiple tissue types, each specialized for different functions, but all working together in aqueous environments. The development of a heart required the coordination of muscle cells, nerve cells, connective tissue cells, and blood vessel cells, all communicating through chemical signals dissolved in tissue fluids. The formation of kidneys required the precise organization of filtering cells, transport cells, and regulatory cells into structures that could process thousands of liters of aqueous body fluid each day. Your own development from fertilized egg to adult human followed developmental programs that were established billions of years ago in the first multicellular organisms. The same basic mechanisms of cellular communication, differentiation, and morphogenesis that pattern your body during embryonic development were first perfected in ancient aqueous environments where the challenges of multicellular coordination were initially solved. The emergence of nervous systems represented a quantum leap in the sophistication of multicellular organization, creating specialized aqueous communication networks that could process information and coordinate responses across entire organisms. Neurons evolved as living wires that could carry electrical signals through aqueous cellular environments, using ion channels and neurotransmitters to convert chemical signals into electrical impulses and back again. Your thoughts right now are the product of electrochemical processes occurring in the aqueous environment of your brain cells, descendants of the first neural networks that evolved in ancient aquatic organisms. Homeostasis, the maintenance of stable internal conditions despite changes in the external environment, became possible through the evolution of regulatory systems that could monitor and adjust the aqueous chemistry of multicellular organisms. Feedback loops involving sensors, control centers, and effectors all operated through chemical signaling in aqueous body fluids, creating biological thermostats that could maintain optimal conditions for cellular function. Your body temperature, blood sugar levels, and pH balance are all regulated through descendants of these ancient aqueous control systems. The development of immune systems created sophisticated defense networks that could distinguish between self and non-self while operating entirely in aqueous environments. White blood cells evolved as mobile defenders that could patrol through aqueous body fluids, recognizing foreign invaders and coordinating defensive responses through chemical communication. The antibodies that protect you from infection are water-soluble proteins that can bind specifically to foreign molecules, neutralizing threats through the same principles of aqueous molecular recognition that first evolved in primitive multicellular organisms. Sexual reproduction in multicellular organisms required the evolution of specialized reproductive systems that could produce gametes, facilitate fertilization, and support embryonic development, all in carefully controlled aqueous environments. The development of internal fertilization created protected aqueous spaces where sperm and eggs could meet and embryos could develop using the same water-based chemistry that had sustained their ancestors in the ancient ocean. Your own reproductive system maintains these aqueous environments with remarkable precision, creating optimal conditions for the next generation. Regeneration, the ability to repair or replace damaged body parts, demonstrated the remarkable plasticity of aqueous biological systems. Some organisms could regrow entire limbs or organs by reactivating developmental programs and recreating the chemical gradients and cellular communications that had originally patterned those structures. Stem cells, those remarkable cells that can differentiate into multiple cell types, maintain their potency in specialized aqueous microenvironments called niches, where chemical signals keep them ready to replace damaged tissues. The evolution of aging and programmed cell death created mechanisms for managing the life cycle of cells within multicellular organisms. Apoptosis-controlled cellular suicide occurred through carefully regulated changes in cellular water balance and the activation of specific enzyme systems that could dismantle cellular components without harming neighboring cells. This process allowed multicellular organisms to eliminate damaged or unnecessary cells while maintaining overall organismal health. Social behaviors in multicellular organisms often involve the coordination of activities across multiple individuals through chemical communication in aqueous environments. Pheromones dissolved in water or carried by aqueous body fluids could coordinate mating behaviors, territorial displays, and cooperative activities. Even the complex social behaviors of modern animals rely on chemical signaling systems that evolved from these ancient aqueous communication networks. The development of learning and memory required neural systems that could modify their connectivity and response patterns based on experience. These neuroplastic changes occurred through alterations in the aqueous chemistry of synapses, the specialized connections between nerve cells, where chemical neurotransmitters carry information from one cell to another. Your ability to learn new skills, form memories, and adapt to changing circumstances depends on the same aqueous neurochemical processes that first evolved in the nervous systems of ancient multicellular organisms. Cancer, the uncontrolled growth of cells that have lost their normal regulatory constraints, represents a breakdown in the aqueous communication systems that normally coordinate cellular behavior in multicellular organisms. Understanding cancer requires understanding how cells normally receive and respond to chemical signals in their aqueous environment, and how these communication systems can now function. Modern cancer treatments often work by restoring normal aqueous signaling or by disrupting the abnormal chemical communications that support tumor growth. In the grand choreography of planetary systems, water emerged as Earth's master climate conductor, orchestrating temperatures across billions of years with such precision that life could not only survive, but flourish in the narrow band of conditions that complex chemistry requires. This wasn't mere coincidence, but rather the expression of water's extraordinary physical properties scaled up to planetary dimensions, creating a global thermostat that has maintained habitable conditions through cosmic seasons that would otherwise have sterilized our world. Picture water as the planet's thermal memory, capable of absorbing enormous amounts of heat energy without dramatic temperature changes due to its exceptionally high specific heat capacity. When the young sun was about 30% dimmer than it is today, Earth's vast oceans prevented the planet from freezing solid by storing and redistributing the available solar energy with remarkable efficiency. Each water molecule in those ancient seas could absorb and hold thermal energy equivalent to 5 times what most other substances could manage, creating a planetary heating system that kept the surface warm enough for liquid water to persist. The formation of ice created an entirely different, but equally important temperature regulation mechanism. Unlike virtually every other substance in nature, water becomes less dense when it freezes, causing ice to float rather than sink. This seemingly simple property had profound planetary consequences. Ice formed an insulating blanket on top of water bodies, protecting the liquid water below from freezing solid even during severe ice ages. Without this unusual behavior of water, early Earth might have experienced complete freeze-over events that would have eliminated the liquid water necessary for continued biological evolution. Evaporation and condensation created the planet's most sophisticated heat distribution system, carrying thermal energy from warm regions to cool ones through the constant circulation of water vapor in the atmosphere. When water evaporates from tropical oceans, it carries enormous amounts of latent heat energy that is released when the water vapor condenses into clouds and precipitation in other regions. This atmospheric heat pump, powered entirely by water's phase transitions, moderates global temperatures and prevents the extreme temperature variations that would otherwise make most of the planet uninhabitable. The evolution of life itself became an integral component of water's planetary temperature regulation system through the development of biological feedbacks that could respond to temperature changes. When global temperatures rose, increased evaporation created more clouds, which reflected more sunlight back to space, creating a cooling effect. When temperatures dropped, reduced evaporation meant fewer clouds and more solar heating reaching the surface. These cloud feedback systems, mediated entirely by water's phase transitions, created automatic climate control mechanisms that helped maintain habitable conditions. Ocean currents emerged as global conveyor belts for heat transport, moving warm water from equatorial regions toward the poles while returning cold water to the tropics in a planetary circulation system powered by water's thermal properties. The Gulf Stream, which keeps Western Europe much warmer than it would otherwise be, is just one example of how water's ability to store and transport heat energy creates regional climates that differ dramatically from what solar heating alone would produce. Your local climate today is shaped by these ancient patterns of oceanic heat transport. The carbon cycle became intimately linked with water's temperature regulation capabilities as atmospheric carbon dioxide dissolved in ocean water, removing this greenhouse gas from the atmosphere when global temperatures rose and releasing it when temperatures fell. Cold water can dissolve more carbon dioxide than warm water, creating a natural thermostat that helps stabilize global temperatures over geological time scales. The ocean's capacity to store carbon dioxide, a direct consequence of water's chemical properties, has buffered Earth's climate against extreme temperature swings for billions of years. Weathering processes, in which atmospheric carbon dioxide dissolved in rainwater slowly breaks down rocks and removes CO2 from the atmosphere, created long-term climate feedbacks that operate over millions of years. When global temperatures rise, increased evaporation and rainfall accelerate weathering, removing more carbon dioxide and creating a cooling effect. This silicate weathering thermostat, mediated by water's ability to dissolve both carbon dioxide and minerals, has prevented Earth from experiencing the runaway greenhouse heating that sterilized our neighboring planet Venus. The development of photosynthetic organisms created new dimensions in water's climate regulation capabilities as these living systems began using water molecules as a source of electrons for converting atmospheric carbon dioxide into organic compounds. This biological carbon fixation, powered by solar energy but mediated by water chemistry, created a living component of the global thermostat that could respond much more rapidly to temperature changes than purely physical processes. When global temperatures rose, increased photosynthetic activity removed more carbon dioxide from the atmosphere, creating cooling feedbacks that helped stabilize the climate. Seasonal cycles, driven by Earth's orbital variations, but mediated by water's thermal properties, created rhythmic patterns of heating and cooling that became fundamental to biological evolution. The thermal mass of large water bodies delayed seasonal temperature changes, moderating the climate and creating more stable environments for life to evolve complex physiological and behavioral adaptations. Your own seasonal biology, from vitamin D synthesis to circadian rhythm adjustments, reflects evolutionary adaptations to these ancient water-moderated seasonal cycles. Ice ages, those dramatic climate oscillations that punctuated the last few million years of Earth's history, demonstrated water's capacity for both climate regulation and climate transformation. The growth and retreat of continental ice sheets created powerful feedback loops that amplified or dampened climate changes through water's unique properties. Ice albedo feedback, where white ice surfaces reflected more sunlight than dark ocean surfaces, could rapidly cool the planet once ice began forming, while the eventual melting of ice sheets could just as rapidly reverse the cooling trend. The atmosphere's water vapor content became a powerful amplifier of climate changes through water's role as the most important greenhouse gas. As global temperatures rise, warmer air can hold more water vapor, which traps more heat and creates further warming. But this same water vapor also creates more clouds, which can reflect sunlight and create cooling effects. The balance between these competing feedbacks, all mediated by water's phase transitions and radiative properties, determines how sensitively Earth's climate responds to other influences. Regional climate patterns emerge from the interaction between water's thermal properties and geographical features, creating diverse climate zones that supported different types of ecosystems and evolutionary adaptations. Monsoons, those seasonal wind patterns that bring life-giving rain to billions of people, arise from the different heating rates of land and ocean surfaces. El Niño and La Niña cycles, which affect weather patterns around the globe, result from changes in ocean water temperature patterns in the Pacific. These regional climate systems, all powered by water's thermal behavior, create the environmental diversity that has driven much of biological evolution. Extreme weather events, hurricanes, typhoons, and intense thunderstorms represent water's climate regulation system operating at high intensity, rapidly redistributing thermal energy from warm regions to cooler ones. Through dramatic phase transitions and circulation patterns, these storms, while potentially destructive, serve essential functions in the global climate system by preventing the accumulation of excessive heat in tropical regions and distributing moisture to areas that would otherwise become desert. Human activities have begun to perturb water's natural climate regulation systems through changes in atmospheric composition, land use patterns, and direct modifications of water cycles. Understanding how these perturbations might affect water's ability to regulate global temperatures requires understanding the complex feedbacks between water's physical properties, biological processes, and atmospheric chemistry that have maintained habitable conditions throughout Earth's history. The future of Earth's climate depends largely on how water's regulatory mechanisms respond to unprecedented rates of change in atmospheric composition. Will water's natural thermostat be able to buffer the effects of rapidly increasing greenhouse gas concentrations, or will the pace of change overwhelm the stabilizing feedbacks that have maintained habitable conditions for billions of years? Your connection to this planetary climate system is immediate and intimate. Every breath you take, every drop of water you drink, every shower you take participates in the global water cycle that regulates the temperature of the only known planet capable of supporting complex life. You are not separate from Earth's climate system, but rather an integral component of the water-mediated processes that determine the future habitability of our world. Across the vast epochs of deep time, the water cycle emerged as evolution's most persistent teacher, creating and recreating environments that challenged life to adapt, diversify, and innovate. This planetary circulation of water, from ocean to atmosphere to land and back again, wasn't merely a physical process, but rather a dynamic, evolutionary laboratory that shaped every aspect of life's development through billions of years of patient experimentation. Think of the water cycle as a cosmic conveyor belt of opportunity, constantly redistributing not just water molecules, but also nutrients, genetic material, and evolutionary possibilities across the planet's surface. Each cycle of evaporation and precipitation created new environments while destroying others, forcing organisms to either adapt to changing conditions or face extinction. This wasn't harsh selection, but rather gentle, persistent pressure that encouraged biological innovation across unimaginable spans of time. The formation of rivers created the first long-distance transportation networks for both water and life, carving channels through landscapes, while simultaneously creating corridors for genetic exchange between otherwise isolated populations. River systems became evolutionary highways where organisms could migrate, disperse their offspring, and share genetic innovations across vast distances. The salmon in your local streams still follow migration patterns that were established millions of years ago, navigating by chemical cues dissolved in the water that flows through river systems, largely unchanged since their ancestors first evolved these remarkable journeys. Seasonal patterns in the water cycle established the first biological calendars, creating predictable environmental rhythms that organisms could anticipate and exploit. The timing of plant flowering, animal reproduction, and migration behaviors all evolved in response to seasonal variations in water availability, temperature, and day length. Your own seasonal biology, from the tendency to sleep longer in winter to increased appetite in autumn, reflects evolutionary adaptations to these ancient water cycle rhythms that your ancestors experienced for millions of years. The creation of temporary water bodies, seasonal ponds, ephemeral streams, and floodplains generated evolutionary pressure cookers, where organisms had to develop strategies for surviving both aquatic and terrestrial conditions. These environments favored rapid development, efficient reproduction, and mechanisms for surviving dry periods, leading to innovations like drought-resistant seeds, accelerated life cycles, and behavioral adaptations for exploiting brief periods of water availability. Mountain ranges and other geographical features interacted with the water cycle to create rain shadows, isolated valleys, and other specialized environments that became natural laboratories for evolutionary experimentation. Islands of habitat separated by barriers of unsuitable climate became places where populations could diverge and develop unique adaptations without genetic interference from related populations elsewhere. The extraordinary diversity of life in tropical mountain regions reflects millions of years of evolution in these water cycle-generated islands of endemism. The transport of nutrients by water created connections between distant ecosystems, allowing innovations that evolved in one environment to influence evolution in completely different regions. Salmon carrying marine nutrients into freshwater systems, rivers transporting terrestrial organic matter to marine environments, and atmospheric water vapor carrying volcanic minerals across continents all created unexpected evolutionary opportunities by providing novel resources in unexpected places. Groundwater systems created hidden aquatic environments that persisted through surface droughts and climate changes, serving as evolutionary refugia where ancient lineages could survive while surface environments became uninhabitable. Cave-dwelling organisms, many of which have remained essentially unchanged for millions of years, represent evolutionary museums preserved by the stable aquatic environments created by underground water systems. The chemical composition of precipitation changed over geological time as the atmosphere evolved, creating new evolutionary challenges and opportunities. Early rainfall was highly acidic due to atmospheric compounds that are rare today, favoring organisms that could tolerate low pH conditions, while selecting against those adapted to neutral water chemistry. As photosynthesis changed the atmospheric composition, rainfall chemistry evolved as well, creating new niches for organisms that could exploit these changing chemical environments. Flood cycles created disturbance regimes that prevented any single species from dominating aquatic environments, maintaining the diversity necessary for continued evolutionary innovation. Periodic flooding mixed genetic material between previously isolated populations, reset ecological succession patterns, and created opportunities for rapid evolutionary change in response to altered environmental conditions. The biodiversity of floodplain forests and wetlands reflects millions of years of evolution under these dynamic water cycle disturbance regimes. The development of soil systems through the interaction of water, rock, and biological activity created entirely new evolutionary opportunities for terrestrial life. Soil formation is essentially a water-mediated process where chemical weathering, biological decomposition, and physical transport combine to create the complex aqueous-solid interfaces where most terrestrial life now thrives. The evolution of root systems, soil microbiology, and plant-soil interactions all occurred within these water-created environments. Ice ages, driven by orbital cycles, but expressed through changes in the global water cycle, created repeated evolutionary experiments as glacial advance and retreat dramatically altered the distribution of aquatic and terrestrial habitats. These climate oscillations forced organisms to repeatedly adapt to changing water availability, temperature, and habitat distribution, accelerating evolutionary change and creating the biogeographical patterns we see today. Your own species evolved during the most recent ice age cycles, adapting to environments that alternated between glacial and interglacial conditions. The formation of inland seas, lakes, and other large water bodies created evolutionary laboratories where organisms could adapt to conditions intermediate between marine and freshwater environments. These systems often became centers of rapid speciation as organisms adapted to unique combinations of salinity, temperature, and nutrient availability. The remarkable diversity of cichlid fish in African rift lakes represents evolutionary radiation that occurred within these water cycle-created environments over relatively short geological time periods. Wetlands became evolutionary crossroads where aquatic and terrestrial organisms could interact and exchange genetic material, leading to the evolution of amphibious lifestyles and organisms that could exploit both aquatic and terrestrial resources. These environments, created and maintained by specific water cycle conditions, served as stepping stones for the colonization of terrestrial environments and continue to be among the most biodiverse ecosystems on Earth. The water cycle's role in dispersing organisms and their reproductive stages created global patterns of biogeography that influenced evolution on a planetary scale. Spores, seeds, and small organisms carried by atmospheric water vapor could colonize distant environments, creating opportunities for evolutionary radiation in isolated locations, while also maintaining genetic connections between distant populations. The global distribution of many microorganisms reflects millions of years of water cycle-mediated dispersal. Climate change, driven by natural variations in the water cycle over geological time, created evolutionary pressure that selected for organisms capable of adapting to changing environmental conditions. Species that could track their preferred climate zones, adapt to new conditions, or survive in refugia during unfavorable periods were more likely to persist through these changes. The genetic diversity and adaptive flexibility that characterizes modern organisms reflects millions of years of evolution under these dynamic water cycle conditions. Your own evolutionary history is intimately connected to water cycle variations that occurred over the past several million years. The development of human intelligence, social cooperation, and technological innovation all occurred during periods of rapid climate change driven by water cycle variations. Your capacity for cultural adaptation, your preference for living near water sources, and your sophisticated systems for water management All reflect evolutionary adaptations to the challenges and opportunities created by water cycle variability throughout human evolutionary history. Every time you check the weather forecast, you're engaging with the same environmental variability that shaped the evolution of your species and countless others throughout Earth's biological history. And here you are, dear fellow traveler through deep time, carrying within your body the living memory of that ancient ocean where life first learned to exist. You are not merely related to those primordial waters. You are those waters, concentrated and organized into the temporary arrangement of molecules that experiences itself as your consciousness. Every cell in your body is essentially a microscopic ocean, maintaining the same basic chemistry that sustained your ancestors for billions of years in the endless seas of early Earth. When you feel thirsty and reach for a glass of water, you're participating in the most ancient biological imperative, the need to replenish and maintain the aqueous internal environment that is literally more fundamental to your existence than the air you breathe. You can survive weeks without food, days without air, but only hours without maintaining proper water balance. This isn't weakness, but rather a testament to water's absolutely central role in every aspect of your biological functioning. Your blood, that remarkable red river flowing through your body, is essentially ancient seawater that has been refined and concentrated over billions of years of evolution. The salt concentration in your plasma is strikingly similar to the salinity of primordial oceans. And the ionic composition, the specific mixture of sodium, potassium, calcium, and other dissolved minerals, preserves chemical ratios that first established themselves when life was learning to regulate its internal chemistry in marine environments. Consider the water within your cells, your intracellular fluid, which comprises about 40% of your total body weight. This isn't ordinary water, but rather a highly organized aqueous solution where every water molecule participates in the complex chemistry that sustains your life. The water surrounding your DNA helps maintain the double helix structure that stores your genetic information. The water in your mitochondria enables the energy production that powers every thought, every heartbeat, every breath. The water in your ribosomes facilitates the protein synthesis that builds and repairs your body continuously. Your brain, that extraordinary organ reading and interpreting these very words, is approximately 75% water, a concentration that must be maintained within very narrow limits for proper function. The water in your brain tissue isn't just filling space, but actively participating in neural transmission, helping to generate the electrical impulses that constitute your thoughts and memories. When you dream, when you remember, when you imagine the vast oceanic past described in this gentle narrative, you're using aqueous neurochemistry that evolved from the first primitive nervous systems that developed in ancient seas. The water that flows through your kidneys each day, nearly 180 liters of it, represents one of evolution's most sophisticated water management systems. These remarkable organs filter your entire blood volume dozens of times per day, precisely regulating the composition of your body's aqueous environment while conserving precious water and eliminating waste products. Your kidneys are essentially biological distillation systems that maintain the ancient oceanic chemistry within your body while adapting to the demands of terrestrial life. Your digestive system creates a series of carefully controlled aqueous environments where food is broken down and nutrients are absorbed using the same basic chemistry that operated in the primordial ocean. Your stomach acid, your pancreatic enzymes, your intestinal secretions, all of these digestive fluids are precisely formulated aqueous solutions that enable the chemical transformations necessary to extract energy and building materials from food. You are quite literally digesting food in internal oceans that preserve the chemistry of life's birthplace. Even your bones, which seem so solid and permanent, are actually dynamic aqueous systems where water comprises about 30% of the total weight. Bone tissue is constantly being broken down and rebuilt through processes that occur in aqueous solution. With specialized cells using water-based chemistry to dissolve old bone mineral and deposit new crystalline structures. The calcium and phosphorus in your skeleton were originally dissolved in ancient seawater before being concentrated and organized by billions of years of biological evolution. Your respiratory system maintains the delicate balance between gas exchange and water conservation that first evolved when life moved from aquatic to terrestrial environments. The moist surfaces of your lungs, where oxygen enters your bloodstream and carbon dioxide is removed, must remain hydrated to function properly. Every breath you take involves the evaporation of precious water, and your body has evolved sophisticated mechanisms to minimize this water loss while maintaining efficient respiration. You are essentially breathing with internal gills that have been modified for air breathing while preserving their aquatic chemistry. The lymphatic system, your body's secondary circulatory system, creates an additional aqueous network that collects fluid from your tissues and returns it to your bloodstream, while also serving as a highway for immune system cells. This clear, colorless fluid that most people never think about is actually a crucial component of your internal ocean, ensuring that your cells remain properly hydrated while also providing a medium for immune surveillance and response. Your lymphatic vessels are carrying the same kind of aqueous solutions that once bathed the first multicellular organisms in ancient seas. Your skin, the boundary between your internal aqueous environment and the external world, is itself about 70% water and serves crucial functions in water regulation. Through sweating, your skin uses water's remarkable capacity for evaporative cooling to regulate your body temperature, essentially recreating the cooling effect of ocean evaporation on a personal scale. The sebaceous glands in your skin produce oils that help prevent excessive water loss, while specialized sensory receptors detect humidity levels and help your body adjust its water balance accordingly. The water in your muscles, about 75% of muscle tissue weight, enables the contraction and relaxation that produces all movement. The sliding filament mechanism that allows your muscle fibers to shorten and lengthen occurs entirely in aqueous solution, with ATP providing energy through hydrolysis reactions that split water molecules. When you walk, when you reach for something, when you smile, you're using molecular machinery that evolved in aquatic environments and still requires aqueous chemistry to function. Your reproductive system preserves some of the most ancient connections to aquatic life. The amniotic fluid that surrounded you during your embryonic development recreated the oceanic environment where your earliest ancestors lived. Your sperm cells, if you produce them, swim through aqueous fluids using flagella that move through the same basic mechanisms that propelled the first modal cells through primordial seas. The very act of human reproduction requires aqueous environments and uses biochemistry that traces its origins to the first sexually reproducing organisms in ancient oceans. Even your body's waste elimination systems reflect your aquatic heritage. Your urine is essentially a concentrated version of seawater that has been processed to remove valuable substances while eliminating toxic waste products. The composition of your tears closely resembles dilute seawater, and the act of crying may be an evolutionary remnant of salt-excreting mechanisms that helped early marine organisms regulate their internal chemistry. Every bodily fluid you produce carries the chemical signature of your oceanic origins. Your circadian rhythms, those internal biological clocks that regulate sleep, hormone production, and countless other physiological processes, evolved from tidal rhythms that originally synchronized life with the regular oscillations of ancient seas. Your tendency to feel most alert during certain hours and sleepy during others reflects biological timing systems that were calibrated to oceanic cycles, billions of years before the first clock was ever built. The very water molecules in your body right now have been cycling through the global water system for billions of years. Some of the hydrogen atoms in your cellular water may have been present at the formation of the solar system itself. The oxygen atoms were forged in the hearts of ancient stars that died long before Earth existed. You are quite literally made of recycled stardust and ancient seawater. organized temporarily into the pattern that experiences itself as your life. When you drink water, when you swim, when you stand in the rain or beside a lake, you're participating in the same planetary water system that gave birth to life and has sustained it through nearly 4 billion years of continuous evolution. You are not separate from this system, but rather its most recent and perhaps most conscious expression, a universe becoming aware of its own watery nature through the temporary arrangement of molecules that is your thinking, feeling, remembering body. As we drift toward the end of our gentle journey through deep time, it's worth pausing to contemplate the extraordinary truth that has emerged from our exploration. Water is not simply the medium in which life happened to arise, but rather the very essence of life itself, the fundamental substance that connects every living thing across space and time in an unbroken chain of aqueous continuity, stretching back to the very beginning of biological possibility. From the first tentative chemical experiments in primordial seas to the complex consciousness that now contemplates its own origins, water has been the constant companion, the patient teacher, the enabling medium for every innovation, every adaptation, every evolutionary leap that has occurred in the 4 billion year story of life on Earth. There is no life without water, not because organisms happen to need it, but because life is water. Water that has learned to organize itself, to replicate itself, to think about itself. Consider the profound unity this reveals across all living things. The virus that seems so different from complex organisms, the bacteria that appear so alien from multicellular life, the plants that seem so removed from animal existence, the vast trees that appear so unlike tiny insects. All of these diverse expressions of life share the same fundamental aqueous chemistry. All use water as their solvent, their transport medium, their reaction environment. All maintain their organization through the same hydrogen bonding networks that give water its unique properties. All depend on the same principles of osmosis, diffusion, and chemical equilibrium that first operated in the ancient ocean. The evolutionary tree of life is not merely a metaphorical tree, but rather a flowing river of aqueous chemistry that has branched and diversified while maintaining its essential liquid nature. Every branch on this tree, from the smallest microbe to the largest whale, from the simplest algae to the most complex human brain, shares the same aqueous foundation. The differences between species are variations on the same fundamental theme: water organizing itself into increasingly sophisticated patterns of chemistry and behavior. Water's role as the universal solvent means that every biological molecule, every enzyme, every gene, every protein that has ever existed has been dissolved, suspended, or organized within aqueous environments. The entire molecular library of life, all the genetic codes, all the biochemical pathways, all the structural innovations that evolution has produced, exists within the aqueous medium that connects all organisms across evolutionary time. You share this chemical heritage not just with your human ancestors, but with every form of life that has ever existed. The cycles of birth, growth, reproduction, and death that characterize all living things are essentially water cycles, processes by which aqueous chemistry organizes itself temporarily into individual organisms before dissolving back into the larger flow of biological water that connects all generations. Your own life cycle, from the aqueous environment of embryonic development through the water-dependent processes of growth and aging, participates in the same cosmic circulation of organized water that has sustained life for billions of years. The intelligence with which you comprehend these connections is itself a water-based phenomenon, the result of aqueous neurochemistry that evolved from simpler, water-based information processing systems that first appeared in primitive organisms billions of years ago. Your thoughts about water are water thinking about itself, the universe becoming conscious of its own liquid nature through the temporary organization of H2O molecules into the pattern that experiences itself as your awareness. The search for life elsewhere in the universe always begins with the search for water, not because scientists lack imagination, But because they understand this fundamental truth, where there is liquid water, there is the possibility of the aqueous chemistry that underlies all known life. The recent discovery of subsurface oceans on Jupiter's moons, the evidence for ancient water flows on Mars, the detection of water vapor in the atmospheres of distant planets, all of these discoveries excite us because they suggest the possibility that water's patient. Chemistry might be conducting the same experiments in organization and complexity that produced life on Earth. The environmental challenges facing our planet today can be understood most clearly through the lens of water's central role in maintaining the conditions necessary for complex life. Climate change, ocean acidification, pollution, and habitat destruction are all fundamentally disruptions of the aqueous systems that have sustained life for billions of years. Understanding our impact on these systems requires appreciating the deep time perspective of water's role in evolution and recognizing that we are not separate from these aqueous processes, but rather recent participants in them. Conservation efforts, sustainable development, and environmental protection all take on deeper meaning when understood as attempts to preserve the aqueous conditions that enable the continued evolution of life on Earth. Protecting watersheds, maintaining ocean health, and ensuring access to clean water are not just practical necessities, but acts of reverence for the liquid medium that gave birth to consciousness itself and continues to sustain it in every moment of every day. The water you will drink tomorrow may have fallen as rain on dinosaurs, flowed through the bloodstreams of ancient mammals, nourished the first flowering plants, or supported photosynthetic bacteria in primordial seas. Every drop carries within it the history of life on Earth, the memory of every organism that has ever lived, the potential for every form of life yet to evolve. When you drink water mindfully, you are communing directly with the deep history of life itself. The technologies we develop for managing water resources, for purifying contaminated water, for recycling and conserving this precious substance, are extensions of the same water management innovations that evolution has been developing for billions of years. Biomimicry, learning from natural systems, often means learning from the sophisticated ways that organisms have evolved to work with water's unique properties. The most advanced water treatment systems are still crude compared to the elegant efficiency of a single living cell managing its aqueous chemistry. The spiritual and philosophical implications of water's role in life extend far beyond science into the realm of meaning and purpose. Many of the world's wisdom traditions have recognized water as sacred, not as primitive superstition, but as intuitive recognition of water's fundamental role in sustaining life. The reverence for water found in indigenous cultures, the ritual significance of water in religious ceremonies, the universal human attraction to lakes, rivers, and oceans all reflect deep appreciation for water's life-giving properties. Your personal relationship with water, how you think about it, how you use it, how you appreciate it, is itself part of the ongoing evolution of consciousness on a water-rich planet. Every moment of gratitude for water's availability, every act of water conservation, every effort to protect aquatic environments contributes to the collective human awakening to our fundamental dependence on and identity with the aqueous processes that sustain all life. The future of life on Earth depends on maintaining the delicate aqueous balance that has enabled billions of years of evolutionary innovation. This requires not just technical solutions, but also a fundamental shift in consciousness, recognizing that we are not separate from nature, but rather nature becoming aware of itself. Water achieving consciousness and beginning to take responsibility for maintaining the conditions that enable its own continued evolution. As you prepare for sleep tonight, surrounded by the gentle sounds of your own breathing, your own heartbeat, your own cellular metabolism, all of which are fundamentally aqueous processes, remember that you are participating in the same planetary circulation of water that has sustained life since its very beginning. You are not just made of water. You are water that has temporarily learned to think, to feel, to wonder about its own existence. The story of water creating life is not a story from the distant past, but a continuing narrative in which you are both character and author, both product and producer. Every decision you make about how to live, how to treat the environment, how to relate to other forms of life, influences the future chapters of this ancient aqueous story that began in primordial seas and continues in the flowing streams of your own consciousness. And so, dear fellow traveler through the vast ocean of time, we arrive at the gentle shore of understanding. We've drifted together through 3.8 billion years of liquid magic and discovered that water is not simply the substance in which life happened to arise, but rather the very essence of existence itself, the patient teacher, the generous provider, the constant companion that has nurtured every innovation in the grand story of biological evolution. Along the way, we've seen that the story of water creating life is not just ancient history, but a continuing narrative that flows through every cell in your body at this very moment. From those first tentative chemical experiments in primordial seas to the complex thoughts flowing through your mind right now, water has been the enabling medium for every step of this extraordinary journey from simplicity to consciousness. So let me ask you once more, gently as I did at the start, where are you listening from tonight? And as you rest there, can you feel the quiet circulation of your own internal ocean, the gentle ebb and flow of aqueous chemistry that connects you directly to those ancient seas where your story began? Perhaps you're near water now, hearing the soft sound of rain, the distant murmur of a stream, or simply the rhythmic whisper of your own breathing. All of these sounds are water, singing the same ancient song that has sustained life since the very beginning. If you found any peace in this quiet expedition through liquid time, I'd be ever so grateful if you'd like the video. Maybe subscribe to join our gentle community of cosmic dreamers, and share this story of aqueous wonder with someone else who might need to remember their connection to the flowing heart of existence. Water's patient chemistry has been writing the story of life for nearly 4 billion years. And that story continues in every drop you drink, every tear you shed, every thought that flows through your wonderfully watery brain. You are not separate from this ancient process, but rather its most recent and perhaps most conscious expression, the universe, learning to appreciate its own liquid nature through the temporary arrangement of H2O molecules that experiences itself as your awareness. Sleep well, fellow droplet in the vast ocean of being, wherever and whenever you are. May your dreams flow as gently as the primordial currents that first carried life across the threshold from chemistry to consciousness. And may you wake refreshed by the same aqueous grace that has sustained every organism that has ever lived on this precious, water-blessed world.